So it looks like we have everybody just joining right now. We'll give it about a few more seconds till everybody gets in. Great. Well, my name is Dave Block. I'm the chair of the Department of Viticulture and Enology at UC Davis, and I'd like to welcome all of you to the program on healthy vineyard soils. First of all, this is our first major extension program of the year, and I'd like to take this opportunity to acknowledge that 2020 was certainly a year that none of us will forget, between COVID and smoke and all the other things going on in the industry. So I'd like to say that we understand that it was a rough year for you, it was a rough year for the university, and let's hope that moving forward in 2021, things get considerably better. The programs that we put on would not be possible without partnership from our partners in our Extension Industry Relations Partnership Program. And you can see them behind me on the screen. These companies and people have committed to partnering with us for 5 years. Some of them have been with us for 5 years already. And some of them have even decided to partner with us again for another 5 years, even before their first 5 years is up. What this partnership allows us to do is to bring you programs not only on campus, but throughout the state when we're not in the middle of a pandemic. And as we found out with COVID last year, it allows us to adapt very quickly to the environment we're in. So it allowed us to put all of our programs online, which over the course of the year reached 6,000 stakeholders, including probably many of you, between the programs like Office Hours with Dave and Anita, our virtual on the road events, all our information on smoke and smoke impact as the fires were happening, including things like the movie that we made to talk about how to do a bucket fermentation. All those things, ability to react to those quickly and provide you with the information that you need, comes from our partners. And so if you're up on that screen, or your company is, we thank you very much for continuing to partner with us. If you would like to be, please contact any of us about becoming a partner. We'd like to include as many as possible to expand what we do to get you the information that you need. So with that, I'd like to say that we have a really great program over the next 2 days. I'd like to thank Con Curterall for putting together this program and all the speakers. I'd like to thank Karen Block and Caroline Furman for helping to organize the program this morning. Without their kind of support, we can't do the programs that we do. And with that, I think it's time to begin the program, and I'll turn it back to Con to actually introduce the program further. So with that, here's to a happier and healthier 2021 to all of you and your family. Con.
Thank you, Dave. This program was a brainchild of myself and Christina Lascano, who's the keynote speaker for this event with our other speakers. We were approached by Napa Valley growers about a year and a half ago to study soil health in detail. So we had a meeting at Oakville Station. I'm a physiologist, so this is not my forte. So Christina put together a wonderful project to study the effects of soil health, how to manage these. And then she will kick off the program. She's in the Department of Land, Air, and Water Resources as a professor. She teaches classes there as well as doing research, advising graduate students. Prior to that, she was at Cal Poly teaching soil science. So without further ado, I'll turn it over to Christina, and I'll turn off my camera and mute myself.
Thank you, Ken, for the nice introduction. I'm gonna start sharing my screen and my presentation. So as Ken said, my name is Cristina Lazcano, and I'm an assistant professor of soil ecology at the Department of Land, Air, and Water Resources at UC Davis. Today, in this presentation, I'm going to talk about how to integrate the principles of soil health into grapevine production in California. And so if you're here, it's because you have an interest in soil health. And over the last years, there has been a lot of talk about the importance of soil health, not just by scientists, but also by industry. And that's because there's more and more data that are coming out that is showing that soils are really important, not just for producing crops, but also for other important functions like regulating the quality of the environment, regulating air quality, and regulating the climate in the long term by sequestering carbon. I'm gonna start by showing you what's the definition, the very general basic definition of soil health. Soil health, when we talk about soil health, we talk about the capacity of soils to function as vital living ecosystems to support the life of plants, animals, and humans. So basically, a soil that is healthy, it's a soil that functions and that provides services. So this is a relatively new concept, but it's based on a not such a new concept, that is the concept of soil quality that you probably are more familiar with. But this concept of soil quality is slightly different from the concept of soil health. When we talk about soil quality, we're talking mostly about like static properties of soils. It's more like a snapshot of soil properties in time. So we're talking about what's the fertility of a soil at a specific time, what's the NPK, right? The content of nutrients. What is the texture of that soil? So it's more of a static concept. When we talk about soil health, on the other hand, we talk about more of the processes that the soil is supporting. So rather than talking about the content of nutrients, we're talking about the capacity of the soil to supply nutrients to the plants. So it's the turnover of nutrients from organic matter into microbes, into the soil solution, and into the plants, right? So this is a dynamic concept. It involves the functioning of the soil. So here's another definition for you that it's a bit more scientific, and that says that a healthy soil ecosystem is a system with integrity of nutrient cycles, energy flows, stability, resilience to disturbance or stress. It's already reflecting that soil is a living system and that it has a specific set of functions and resists stress. So another idea that it's enclosed into this definition of soil health is that soils are living systems. And this is also something relatively new. So for most of the history of soil science, we were talking about the chemical or the physical properties of soils. So we know a lot about um, soil nutrients and cation exchange capacity or soil texture and other physical properties of soils. But up to a few years ago, we didn't know so much about soil biology. And now, because we have molecular techniques, we are starting to learn that soils are actually extremely diverse. Soils are amongst the most diverse ecosystems on Earth, and virtually all life kingdoms are represented in soils, from bacteria, fungi, archaea, all the way to vertebrates like moles, going through um, the infamous nematodes, but also earthworms and other things that are less known, like collembola. And you have them here. I'm gonna use my uh, laser pointer. So this is collembola. This is a tardigrade. So there's extremely diverse array of all sorts of sizes, shapes, and colors, right, of soil animals. The most interesting thing, though, is that these organisms are not isolated from each other. They are in constant interaction, and it's that interaction that regulates the processes that support the health of soils. And for example, a very common type of interaction in soil is what we call trophic interactions and involve organisms feeding on each other. That's represented in what it's called a soil food web. And here we have a simplified food web that is showing how organisms in soils are feeding on each other. So for example, we got organic matter, right, and plants, and then we have organisms that feed directly on organic matter and plants like bacteria and fungi. We have then microbial grazers like nematodes, for example, that can feed on fungi or on bacteria. And then we have predators and higher level predators that feed on those microbial grazers and so on. So in this way, the energy and the matter that is synthesized by plants through photosynthesis is transferred all the way into higher trophic levels. So these trophic interactions facilitate the transfer of energy and matter in the soil. These trophic interactions therefore support the functions and properties of a healthy soil. And which are those properties and functions? So here you have a simplified schematic showing a summary of the properties of a healthy soil. They are classified by color depending on the category. So we have physical, agronomic, chemical, and environmental properties. In green, we have the agronomic properties. So a healthy soil, it's supposed to support high yields, but not only that, it's supposed to support high quality. And this is especially important in wine grapes, right? We want a specific chemical profile. If we talk about biological properties, here in orange, a healthy soil supports a diversity of organisms which are also active. Talking about environmental properties, a healthy soil stores carbon and also reduces greenhouse gas emissions. In terms of chemical properties, we have them here in red. A healthy soil needs to supply nutrients to the plant, but also should be able to retain the nutrient and not lose nutrients through leaching, for example. And in terms of physical properties, a healthy soil needs to store water, but also not too much. So a healthy soil needs to have good water infiltration and drainage. So that's a lot to ask, right? All right, so how can we get to have a healthy soil? There is 3 basic principles of healthy soil management. Those are, first of all, increasing soil organic matter. And in the context of vineyard management, you can do that by applying compost, for example. The second one is reducing disturbance to the soil. An example of a management practices that reduces disturbance is reducing till or using no-till. And the third one is increasing diversity and maintaining a living plant cover. And a very good example for doing that is cover cropping, right? We are introducing a diversity of plant species into the vineyard. That cover cropping actually also contributes to increasing soil organic matter, right? The same as compost. So there is a set of a variety of practices that one can use in a vineyard to support soil health. For the purpose of this presentation, I'm going to focus on a couple of examples on how management affects soil health. And I'm going to be just focusing on compost application and how it increases soil organic matter. Organic fertilizers like compost are a direct input of organic matter to the soil, and so therefore they increase soil carbon directly, has benefits for carbon sequestration and long-term climate regulation, right? So that's a direct benefit. But also soil organic matter has a variety of benefits for other soil properties, like, for example, biological soil properties, because organic matter compost feeds the food web. Compost also improves physical properties of soil and chemical properties of soil by supplying nutrients, increasing cation exchange capacity, etc. So in the next slides, I'm going to show an example of a study that we did a couple years ago, in which we studied how compost application it's affecting the soil food web. This study was carried out by the master student Holly Denniston Sheets, which is now working at the Citrus Board, and in collaboration with researchers from UC Davis and from Cal Poly San Luis Obispo. It was done at Niven States, Niven Family Wine States in San Luis Obispo, and was supported by the California Agricultural Research Institute of the California State University. So the objective of this study was to assess the effects of fertilizer inputs, either organic or inorganic, on soil health by using nematode food web as proxies. And you will say, why nematodes? You probably know nematodes as like the bad guys. Nematodes have a very bad reputation because they cause a lot of damage, right, for crops. Some of the nematodes are plant parasites, but there's actually a wide variety of nematodes in soils, a great diversity, and a lot of them are actually beneficial. So we got, the interesting thing about nematodes is that besides the plant parasites, nematodes are actually represented in all of the trophic levels of the food web. So we have nematodes that feed on plants, those are the bad guys, but we also have nematodes that feed on bacteria and fungi. Those are bacterivores and herbivores and are beneficial because they support nutrient cycling. And we also have nematodes that are predators and nematodes that are omnivores, and they would eat anything, right? So by looking at nematodes, we can get a very good idea on how the whole structure of the food web is changing with different management strategies. Nematodes can be very easily classified into trophic groups by looking at the morphology of their mouthpieces. And so you have an example in this picture. You see here, and this is something that is probably familiar to you. This is a nematode, and this is the mouthpiece. This is what the nematode uses to feed. And in this case, we're seeing a plant herbivore. And we know that because it has that little stylet there that is like a needle that they use to puncture the plants and suck the juices of the plant, right? So in the same way that we can identify this as a herbivore, we can identify bacterivores, fungivores, predators, etc., because they change their mouthpieces. Overall, nematodes are quite sensitive to disturbance, and they are correlated with beneficial functions in the soil. So they're really good for studying ecosystem processes. And this is what we did in this study. What I'm gonna show you now, it was part of a bigger study where we looked at the effects of weed management strategies and also cover crops on soil health. But again, for the purpose of this presentation, I'm just going to focus on the fertilizer trial. So in this case, we had a control that didn't receive any fertilizer. And then we had two treatments: synthetic fertilizer applied under the vine and as foliar sprays, and organic fertilizer that was poultry manure compost applied under the vine. All the treatments were replicated 6 times and arranged in the field following a complete randomized design. So the arrangement of the treatments, it's represented here in the center of this picture. This is an aerial view of the field. So you can see how the treatments were distributed randomly in the field. Within each plot, each experimental plot, we looked at 2 locations separate, the tractor row and the soil under the vine. And the reason for this, it's because, as you know already, both soils receive very different management. The soil in the tractor row, it's tilled every other day. It usually has a cover crop or some kind of vegetation cover. And whereas the soil under the vine, it's still much more frequently for weed management. It gets water, it receives the fertilizer. So we could expect also quite different effects, right? And different food web structures in the two locations. And so that's actually what we've seen. And this bar chart is showing you the total abundance of nematodes, regardless of their trophic group, right? So we here see that the overall abundance of nematodes was much higher in the soil in the tractor row than in the soil under the vine. Afterwards, we went to look at, all right, so what are the different types of nematodes in each of the locations? And this is what you can see here in these pie charts. On the top, we have the tractor row, and the different colors represent the different trophic groups. And in the bottom, we have the vine row, and again, different colors represent the different trophic groups. So what stands out is that the orange was much more abundant in the vine row as compared to the tractor row. Orange was herbivores. So the abundance of herbivores, the plant parasites, was much higher in the soil under the vine than in the tractor row. The soil in the tractor row had a way more balanced distribution of the different types of trophic groups of nematodes. It had more fungivores, more bacterivores, more predators, and more omnivores than the vine row. Okay, so the abundance and diversity of nematodes is quite different in the soil in the tractor row than in the soil under the vine. The next question is, all right, how does the application of the different fertilizers affect these nematode abundances? And this is what you have here in this slide. The bar charts represent the abundance of the different types of nematodes in the control, which is the no fertilizer treatment, the organic fertilizer, and the synthetic fertilizers. So as you can see here, there were no differences in the abundance of plant parasitic nematodes, although there was a slight trend for organic fertilizers to reduce the amount of plant parasitic nematodes. But what we've seen is that organic fertilizers increase the abundance of fungivore nematodes and bacterivore nematodes, as you can see here in this other chart. So how is this possible? Organic fertilizers are an input of food for the food web, right? So adding organic fertilizers increases microbial biomass, both fungal and bacterial biomass. And that actually increases the abundance of the organisms that feed on them. That's bacterivore nematodes and fungivore nematodes. So organic fertilizers, by constituting a food source for the food web, increases the complexity of the food web, which is an early sign of soil health. In the last part of the presentation, I'm gonna move a little bit into a different aspect of organic fertilizers, which is the environmental benefits and balancing environmental benefits with agronomic outcomes. So as I mentioned before, adding organic fertilizers to the soil represents a direct input of carbon. So organic fertilizers have benefits for carbon sequestration and long-term carbon regulation, all right? And once we add that organic matter into the soil, that organic matter is going to become stabilized. And that's what we call carbon sequestration, right? It becomes somehow locked up in the soil and is not available for microbial breakup. And so it's going to be stable over time. And when it's stable in the soil, it's not released to the atmosphere as CO2. So this chart that looks a little bit complicated, it's actually showing the different ways in which carbon can become stabilized or sequestered in the soil in the long term. So an input of organic matter into the soil is initially unprotected carbon, right? All of it is available for microbes. Out of that general input of organic matter in compost, there will be some of it that it's very recalcitrant and it's very difficult for microbes to break down, right? It's like when you go to a buffet and you have cookies and you have broccoli. Nobody wants the broccoli, right? So that recalcitrant organic matter is the broccoli. There's no microbe that's going to go and get the broccoli. So that becomes immediately stabilized into the soil. It's non-hydrolyzable organic matter that it's going to be biochemically protected soil carbon, right? The rest of the carbon, it's going to be broken down somehow and consumed by microbes. As the carbon is being broken down, the microbes produce residues, and those residues are sticky. They are molecules of organic matter that become sticky and they attach to the mineral particles in the soil. So this is what we see here. This is what we call the physical and chemical protection of carbon. Those sticky products, they can be incorporated into the aggregates, right? And so become part of the structure of the soil. And in that way, when those organic products are locked into the aggregates, they are not available for microbes. So they cannot be broken down. And on some other cases, some of the microbial residues are going to be attached to silt and clay particles and also become stable. They are protected when they are attached to the mineral particles. So overall, there are different types of carbon stabilization in soil. There is biochemical stabilization, and there is physical and chemical stabilization of carbon, all right? This is what happens after we put the carbon into the soil. Now, if you notice in this slide, at the same time as we have carbon being stabilized in the soil, we have CO2 being produced. And this is natural, it's part of decomposition. There's always CO2 being produced, right? The question is, how much of that CO2, of the carbon that it's put into the soil is going to be released as CO2? Can we reduce that? Another important thing to consider is that when we are adding carbon to the soil with organic fertilizers, we're also adding nitrogen. Nitrogen has to be mineralized. Once it's mineralized, it turns into ammonium. And as you know, ammonium is nitrified really, really quick in soils to produce nitrate. And then nitrate can be subjected to what we call denitrification to produce N2, which is returned to the atmosphere. These are all natural processes that occur in soils. The interesting thing is that as these processes are happening, there are certain intermediates to these reactions that are produced and that are also gases, and they are released to the atmosphere. One of those gases is N2O, or nitrous oxide. Nitrous oxide is a greenhouse gas that is 300 times more powerful than CO2. So again, once we put organic matter into the soil, we need to be careful and understand how these two gases are produced, so we reduce the emissions of these gases. Okay, so this could be an actual trade-off to the environmental benefits of using organic fertilizers. So as we add compost to the vineyard, we're adding nutrients, we're adding carbon that is going to contribute to soil carbon and nitrogen pools, and it's going to contribute to the nutrition of the vine. But at the same time, just because of the natural processes of decomposition and nitrification and denitrification in soil, there's going to be a certain amount of the carbon and the nitrogen that is lost as CO2 and N2O. So we need to balance both the environmental and the agronomic benefits, right? And for that, we need a very careful management of a compost application. So the next slides, I'm going to show you a study that we are actually still running at Jaylor Vineyards in Paso Robles, with support from the CDFA and also the ARI, the Agricultural Research Institute of the California State University. This is a study that it's being carried out by the graduate student and CFI, and also in collaboration with researchers from UC Davis and also from Cal Poly, San Luis Obispo. The objective of this study is to determine what rate of compost application is going to maximize the benefits to soil health on one hand, but also reduce environmental impacts, reduce the emissions of greenhouse gases. And here's a little bit of the experimental design of this study. We had 4 rates of compost, a 0 application rate, that was our control, 2, 4, and 6 tons per acre being applied. The compost is broadcasted to the whole vineyard. The treatments are distributed following a randomized block design. So this is how the field trial would look from the air. We have 4 blocks, and within the blocks, we have the treatments, the different rates distributed randomly, right? So in blue, we have the 0 tons per acre. The green is the 2 tons per acre. Yellow, it's 4, and orange is 6 tons per acre. The compost that we are applying is a certified organic compost produced at Cal Poly, and it's made of manure and also green waste. And as I mentioned, it's broadcasted to the whole vineyard once a year, and it's usually after harvest, before the first rain in November. Again, same as with the other trial, we studied 2 different functional locations within each plot. We studied the tractor row and also the soil under the vine, just because those 2 locations receive different management. Every year we went and collected, after compost application, we collected soil samples, and also after every management event, we went and sampled gases that we later analyzed in a gas chromatograph in the lab. At harvest, we also assessed yields, and we picked berries for chemical analysis. We analyzed Brix, anthocyanins, phenolics, malic acid, and acidity. And so I'm gonna show you some of the results of this study, and I'll start talking about active carbon, or POXC, that we call permanganate oxidizable carbon. Active carbon, it's a fraction of total carbon that it's actually sensitive to management, and it can be used as an early sign of changes in carbon sequestration. So in this chart, you can see the different treatments, the different rates of compost with the different colors in the tractor row, the vine row, at different soil depths. So we collected soil in the depth interval from 0 to 6 inches, then from 6 to 12 inches, and then from 12 to 24 inches. So as you can see here, there's a very marked difference in depth. So it is the top soil that has the highest amount of carbon, right? And also we saw a very strong difference in that top soil layer between the tractor row and the vine row. It is the tractor row that has the most carbon with difference, right? However, there was no effect of and there was an effect of the compost rate. Higher rates applied increase the amount of active carbon in the topsoil, and especially in the tractor row. The next slide is showing the emissions of the greenhouse gas nitrous oxide. And in this case, we have it broken down by season. And the reason for this is that the different seasons, we have wet season, which is normally winter to spring. And we have dry seasons, which is the growing season from summer to fall. And between the wet and the dry season, nitrous oxide emissions are actually quite different. The wet season triggers more emissions because it is the rain and that actually triggers nitrous oxide to be produced in the soil. So we have the cumulative emissions represented different in the different seasons, all right? So you can see here in the first wet season, we did not see any differences in the different compost treatments on nitrous oxide emissions. Same for the second wet season that we analyzed. But in the dry season, that was 2019, summer to fall, that was the growing season, we saw a difference between the soil under the vine and the soil in the tractor row. The soil under the vine produced more nitrous oxide than the soil in the tractor row. And again, the reason for this is because the soil under the vine receives irrigation, and that irrigation creates water saturation in the soil that triggers the production of nitrous oxide through denitrification. Okay, so in this case, compost doesn't have any effects, but it's the irrigation that triggers the production of nitrous oxide. The next graph shows cumulative emissions of CO2, again, for the different seasons of this study. We have one wet season followed by one dry season, and again, another wet season. So what we've seen here, it's also quite different to nitrous oxide. First of all, there was no effects of compost on CO2. So compost addition did not increase the emissions of CO2. But we did see very strong differences between locations, between the vine row and the tractor row. During the wet season, it was the tractor row that had the highest CO2 emissions as compared to the vine row. This happened for the 2 wet seasons of the study. During the dry season, these relationships changed. It switched, so it was the vine row that had the highest emissions. The reason for those differences between locations, again, it's water. Water in the case of the summer season, because the vine row receives irrigation that stimulates microbial activity, and microbes, as they are active, they respire and they produce CO2. Now, during the wet season, it was the tractor row that produced the most CO2 emissions. And the reason for that is that the tractor row has a cover crop growing in it. And the cover crop also respires, right? So that CO2 is coming from the cover crop. But again, no effects of compost on CO2 emissions. We went to look at the quality of the grapes, and this is data from harvest in 2019. We still have to analyze last harvest. We're working on that. But as you can see here, there was almost no differences with all the compost rates on the different chemical parameters that we analyzed. So neither anthocyanins, phenolics, or acidity were different with the different compost rates. We didn't see effects in malic acid, and we didn't see effects on pH. And there were also no differences in total yields. So overall, compost applications show benefits for soil health in the sense that it increased the amount of active carbon in the soil. And that was especially remarkable in the tractor row and in the top layer of soil. When we looked at any potential trade-offs of compost application by looking at the emissions of CO2 and nitrous oxide, we saw that compost application did not increase the emissions of these greenhouse gases as compared to a soil that did not get compost application. So compost has benefits for soil health, but very little trade-offs for environmental quality. We looked at the quality of the grapes and the yields, and they were not changed by compost application. We expect that these changes with further applications of compost. So we'll see what this year's results are going to be. All right, with that, I'm going to wrap it up, and I think I'm already over time. So thank you very much. And if you have any questions, you can either make them through the chat or you can also send me an email. Thank you.
Okay. All right. I think we have some time for questions. Correct, Caroline? Or
Yes,
did
we do. We have a few minutes.
Yeah,
Perfect.
we have a few minutes, Christina. So we got some questions in our chat.
Great.
So in chat, the first question is compost application and depth. Do they have any advantage compared with a broadcast or a band application under California climate?
Yeah. Yes, we've seen that a lot in different vineyards, that each vineyard has a different application, a different application strategy, right? Most vineyards actually ban the compost under the vine. That's what I've seen the most. Now, in our studies, we've seen that it's the tractor row, the soil between the vines, that it has the highest potential to sequester carbon. And the reason for that is, well, the management of the soil first, but also the fact that cover crops are usually grown in the tractor row. So layering compost application with cover crop has a synergistic effect on carbon sequestration, because your compost is going to be stimulating the growth of the cover crop. And so there's going to be more carbon into the soil than if you just use compost, right? So for in terms of like sequestering carbon for the whole vineyard, I would say that broadcasting is the most efficient, but most managers, most vineyard managers apply under the vine because they use compost as a nutrient supply, right?
Yes.
So it depends on the purpose of compost application.
Um, the next two questions can be combined. What sort of organic fertilizers were used in the Paragon vineyard and at what rates?
So in Paragon, we use composted poultry manure,
Mhm.
and I can't recall now the rates, but I can send them to you. Yeah, and in the case of the Jailor study, we used compost was made with dairy manure and green waste. It was a mix.
Next question is, do conventional inorganic nitrogenous fertilizers contribute as much as organic nitrogenous fertilizers to nitrous oxide production?
That's a very good question. And generally, they can, yes.
Yeah.
But generally so inorganic fertilizers can usually have higher emission rates than organic fertilizers. That's what the literature is showing.
Yeah.
But there is a risk. It depends on the form of organic fertilizer that you're using. So if you use manure, for example, manure has a lot of ammonium and like easily available nitrogen, plus easily available carbon, and that's going to stimulate denitrification rates, both the nitrogen and the carbon. So if you apply manure, you might actually be emitting as much as with an inorganic fertilizer. But compost is more stable. It has lower amounts of easily available nitrogen and carbon. So the nitrification and denitrification rates are slower.
Yeah. Okay. Yeah, if you can please ask your questions in the Q&A rather than the chat. It's a tab under your screen. The other question is, to reduce tillage, would broadcasting compost on the surface without incorporation provide a reduced, similar, or increased benefits in comparison to incorporated or tilled-in fertilizers?
That's a very good question.
Yeah.
So incorporating organic matter, either compost or cover crop, it actually facilitates the mixing of that carbon and the breakdown of that carbon and the stabilization of that carbon in some way. But tilling the soil also breaks down the aggregates that stabilize the carbon in the long term, right? So it's a balance between how much you can stabilize and how much you're going to be destroying of that stable carbon. And that's why maybe certain level of tillage might actually be beneficial as compared to no-till, right? But if you go too far, you're actually breaking more carbon than what you are incorporating by breaking down those aggregates, right? Those mineral and organic associations in the soil.
That's kind of what we saw in Fresno when we were running a trial. I mean, it ended up being like an ashtray in the end. It was so fine. So,
Yeah.
nice.
Yeah,
Caroline,
tillage
how
is
are we doing on time for the next time?
We have about 9 more minutes for
okay.
questions.
Yeah. Okay. I can ask you. I'm going to keep asking you these questions, Christina.
Go ahead. Go ahead.
There's like 10 questions. So, anonymous attendee asks, oh, I already asked that. Since tractor rows have the most diverse nematode populations, doesn't it indicate that vine rows should be allowed to have cover crops in them too? Is the proclivity to clear cultivate in the vine rows not a healthy practice, especially for an organic vineyard?
Yeah. Yes, I mean, generally having a plant cover on the soil is what's going to keep it healthy, right? But then again, we are working with crops, so the business is growing the crops, and then there's decisions that need to be made, and so at some point, then the crop is prioritized, especially when there is a shortage of water or there might be a risk of nutrient competition, and you can know more about that, right? You've been looking at cover crops as well recently,
Yeah.
and competitions between the cover crop and the vine for water. So yeah, there is also the alternative of
Yeah, I mean,
terminating
that's
the
been
cover
a,
crop. Go ahead.
I mean, our biggest issue with cover crops under the vines has been a competition with the target crop, which is the grapevine. And so, I mean, grass species, tall grass species compete very well with the grapevine roots. So we really have not seen many benefits of like barleys, oats, etc., using them as cover crops in vineyards because it just like sucks it up dry. But that's been our experience since 2013, and I have mainly worked in San Joaquin Valley with those. In coastal areas, it might be different. I mean, in Napa, where we have the water table down to about like 9 feet, it might be beneficial to suck this excess moisture out. But that study is still ongoing.
That's right. Yeah, it depends on
So,
exactly on
anyways,
where you're
next
growing.
question. Next,
Or
go ahead.
it depends on where you grow in your grapes, and then there's also different ways of managing that competition, right? And one of the ways of managing that is terminating the cover crop, right before bad break or in early spring, and that's something that actually happens kind of naturally also as things start to get dry as well. But under the vine, yeah, there's irrigation in most of cases, so cover crop will keep growing. Yeah.
Okay. All right. Next question is, does tilling in the cover crop erase its benefits of promoting carbon storage?
Tillage, sorry, go ahead. Can you repeat?
Does tilling in the cover crop reduce its benefits of carbon storage?
Not necessarily. Again, it depends on how much you till. Um, there is a fine balance between using tillage to actually incorporate the cover crop and promote a faster incorporation of the carbon into the soil, and go tilling too much and destroying the structure of the soil so the carbon actually doesn't stay. Tillage does not necessarily um, reduce carbon storage, but it depends on how it's managed. And you know, there's, again, it's very site specific. It depends on the soil that you have. Some soils are, they have extremely high contents of clay, and they store more organic matter just naturally, that some other soils that are very sandy and it's very extremely difficult to accumulate organic matter, right? So the manage of the
That's
two
correct.
types of soil, then it's going to be different. So some heavy clay soils, um, then they need to be tilled at some point, right? So a certain very light level of tillage might actually help incorporate that organic matter while not actually damaging the carbon in the soil. It seems that there is some delay.
Yes. So, anyways, so for questions that we do not get, can answer these in chat. We still, Christina, you got a lot of questions, so that means it was a great talk.
That's
So it
great.
generated a lot of interest.
That's great. I'm just sorry that, you know,
So,
we can't be in person and I can't just get to talk to everybody. But you, hopefully soon. Hopefully we can do these things in person soon.
I'll take one more question, then we will move on. So what are the most relevant traits that growers are looking from organic fertilizers, such as key metrics that you would like to optimize an organic fertilizer standardization?
Okay, well, that's a good question. So there are certain like quality parameters that you need to look for. The compost, if you're using compost, it needs to be produced in a certified facility, and it needs to be stable, so well finished. And so that's usually what I would look for. Some people do composting in house, and that's fine. You just run into the risk of not having the compost completely finished, which it might cause maybe some issues of, you might lose some nitrogen when you apply it. But again, it depends on the mix of organic waste materials that you're using. Then another important thing to look for, especially in wine grapes, is the carbon to nitrogen ratio. Um, because of the problems with nitrogen and vigor, you don't want to apply organic fertilizers that have high contents of nitrogen to wine grapes. So I would be very careful with, obviously, you know, applying um, certain organic fertilizers that might have higher nitrogen content. Those are usually manure-based fertilizers. So composts that have a higher carbon content as compared to nitrogen are usually produced from plant or like green waste, mixed with a little bit of manure as well, but there has to be that mix. And I know that some growers are also using their own winery waste, so grape pomace produced
Yes.
from, yeah, and mixing it with pruning waste as well. That's a very good mix, but it has to be properly composted, because grape pomace, it's very damaging to the soil and to the plants. If it's applied directly, it's very saline, it's phytotoxic. So knowing how to do the compost is important. How
Yeah.
long you need to turn it for, and what kind of properties do you need to look for.
Okay. All right. Well, thank you, Christina. I think we will move on. We do have some more questions to get through in Q&A.
All
So
right.
it was a great talk. And then
Thank you.
we will see Christina again tomorrow in our program. And hopefully we will do these in person very soon.
Hey, Khan, is it possible that maybe Christina then can answer some of those questions while the other speaker is going?
Sure.
Yeah.
Sure.
I will look at the chat and try to address the
It's the
question.
Q&A. Yeah, thank you.
Okay.
Okay. All right. Let me queue up my talk. Hang on. All right. I'm the next speaker. So I will share my screen. Okay. My name is Khan Kurtral. I'm the CE specialist for viticulture at University of California. So I will talk about vineyard site preparation, some practical information for the growers, as we do have a lot of development and redevelopment happening across the state of California. So some of the photos I have, they are coming from our extensive network of farm advisors, colleagues, such as Mark Batney, Rhonda Smith, who recently retired, Glenn McGordy, who recently retired, UCANR Photo Bank, and my colleague Daniele Zachariah in LAWR. We need to prepare the vineyard sites because a lot of the commercial cultivars that we are using are not native to North America or North American soils. And as Christina alluded to it, and as my colleague Andy will talk about, we do have a lot of diseases, as like Andres will talk about. We do have a lot of nematodes. Cindy will talk about viruses. We have a lot of, things that can affect the survivability of these vineyards. And vineyards are perennial plantation systems that are irrigated, trellised. There's very heavy plant protection controls, and they are highly mechanized in terms of how a lot of practices are done. Some factors to consider for vineyard site preparation, the soil factors. The soil factors are a limitation to plant growth. We do have some sites that need to be avoided. And the thing to consider is a fundamental site capacity when we are looking at vineyards. The other thing that we have to worry about is that we are operating in California, and most of the West Coast requires irrigation. The number one thing is quantity of water. Will you have enough? The other thing to consider is the quality of water. If you're dealing with brackish water or a high boron water, how these will impact the operations over time. Probably the most important factor in the end, grapes are a business. You have to consider the production factor. How will this vineyard be farmed? Majority of new vineyards are being planted with the aim to mechanize at least half the cultural operations. On your right, I have a vineyard from Kern County, California. This is a Pinot Gris on 1103P rootstock. These rows are 2 miles long. We are looking for uniform canopies with a consistent yield, and these things have to have acceptable fruit composition to contract specifications year after year, and the soils have to perform to deliver these. Some soil factors to consider. Technically, we are looking for a tilth, a physical suitability to grow the crop. are physical limitations in soils, such as those in a bit. There are chemical limitations, such as lime content of these soils with a high magnesium, fertility, excess or insufficient fertility as it relates to organic matter in soils. And of course, there's the variability of soils, and this is unavoidable, and that will challenge uniform growth in vineyards. Evaluation of soils ideally would start soon after vineyard removal, or as soon as you start negotiating for a piece of land. Usually, evaluation of soils used to be based on a grid-based sampling. This is more cores per management unit and more soil pits per management unit. And this is more of a bulk management strategy, which still works quite well. However, as we developed our methods over the last 30 years, we are using more of a stratified sampling, such as vineyards being sampled proximally. This results in less cores per management unit, less soil pits per management unit, and this is more towards moving towards zonal management in vineyards. Grid-based soil sampling. A grid is laid over the vineyard. A certain number of cores are pulled, and a certain number of pits will be opened. So in this management block, for example, 14 cores were pulled and analyzed, and approximately 6 soil pits were opened to assess the suitability of this site. Sensing-based assessment uses electromagnetic induction sensing. This will usually cost $40 to $60 an acre. What we'll do is generate zones based on electrical conductivity or electrical resistivity, and it will pinpoint where soil cores or soil pits have to be open to do the sampling in these vineyards. However, this is something that needs to be studied further by the grower before delving into this. The sensor-based zoning needs to be measured soon after spring rains. The interpolation of the zones needs to be asked by the grower, because there are spatial dependencies in vineyards, and this needs to be assessed by the service provider. The spatial dependency index will verify the levels of spatial dependency in the data set. Usually a semi-variogram is drawn, and based on the nugget and the sill, which you will get from the semi-variogram, will indicate if there's strong spatial dependency, moderate spatial dependency, or weak spatial dependency. Meaning that if your values are less than 25, you will have a stronger spatial dependency, meaning that the soils are very variable. You need to divide this vineyard into zones. Checklist for adjustments in vineyard soils. Of course, proper sampling, testing, and interpretation of results are essential, such as soil pH, drainage, tiling will be required, amount of organic matter in the soil, gypsum needs, and compost needs will need to be attended to. Chemical properties for vineyard soils, we're usually looking for a pH between 5.5 to 6.8. This is a very wide window, and in Napa, we do have soils that are between 5.5 to 5.7. The vines are performing, okay, so it's not a cause for concern that much. Organic matter, we're looking for a test between 1 to 3%. Phosphorus test, 40 to 50 pounds of phosphorus to the acre. Potassium is probably the most overlooked element when vineyards are being prepared. We're looking for a test of plant available potassium of 250 to 300 pounds to the acre, and magnesium about 200 to 250 pounds to the acre. A lot of these things can be amended before the vineyard is planted, and they do have to be incorporated prior to the vineyard being installed. There's a need for evaluation of vineyard soils, like I'm first photo here. You can see that this is a very interesting soil structure that we see here. As you go down about like soil, you have this impermeable. There are topography challenges. Yeah, this uh, uh, side hill is eroding soon after spring rains. This will need to be terraced. Of course, shallow bedrocks and deep restricted drainages will have limitations of plant growth, and these are all pre-planting decisions that need to be taken care of before the vineyard has been developed. Water infiltration is a constant concern, and these are results of hard pans in the vineyards. And these hard pans will, stop water infiltration. And as this vineyard was assessed after the fact that it was planted, there was a very nice, hard pan layer about 2 feet down into the vineyard soil. There are impermeable clay layers that might happen, and this will more often than not result in poor grapevine performance. In this otherwise fine performing young vineyard, there were spots where even the weeds did not grow, and the cause was an impermeable layer in this vineyard, as you can see. Waterlogged soils are usually caused by heavy clay, poorly drained and aerated soils, and these can be difficult to identify later on in the season. But early in the season, you can see that the vines are not performing quite well. Of course, these zones can be identified via sensing, and these can be tended to as the vineyard matures in this case. Some of the textural problems we see in these vineyards, there might be an abrupt texture interface where you have a nice tilth in the top portion of the soil, but then there might be a sandy clay layer which creates a zone of saturation in these soils. And this abrupt texture interface, as it changes to pure sand, it will limit the proliferation of the roots for a healthy production of grapevines in these poorly prepared vineyards. Lyme chlorosis, sometimes that will happen due to improper rootstock choice. Andy Walker will allude to it tomorrow. A lot of the rootstocks will have some vinifera parentage into it. A lot of the rootstocks will not have enough vinifera parentage in it. Some of the rootstocks will not take to the soils we have in North America as well as they should, and this is a rootstock problem that can be attended to before the vineyard is installed. There are limitations to changing the soils. We can do a lot of practices, deep ripping, of course, gypsum additions, and of course, adding sulfur to change the acidity or pH of these soils as these vineyards are prepared. However, these do need a careful planning and analysis by our professionals that provide these services. Site preparation. Considerations for site preparation, exploit the capacity of vines, matching it to the site vigor and the intended manner of management. Sometimes aerial capture of variability is not at these two variability maps of leaf area index and NDVI captured aerially. These are not indicating that this vineyard is very variable. However, to our eye, it does look very variable because of the coloring schema on these maps. However, as these are delivered to you, make sure you ask for the Cambardella index or the spatial dependency index. As I mentioned previously, these values need to be below action in this case. Agricultural lands are variable in space. Vigor differences are related to the soil's ability to supply water. Yes, even in irrigated vineyards, the soils will have the capacity to supply water later on into the season. In our case, when we studied this in greater the stormwater potential over the course of the season, the vineyard is divided into two zones, and this is consistent over 2 years. And then the plant water status of this vineyard is making a similar map 73% of the time, year over year, meaning that this relationship is stable and it will not change within our lifetimes. But why is this so? And can you sense this and take action towards it? Yes, of course you can. This can be sensed and applied to very large acreages. This work in California has been completed in Sonoma, Napa, San Joaquin, and Tulare counties. Results have always been similar. When we sense soils at two depths between 0 to 75 centimeters, or 75 to 150 centimeters. And this will match if the spatial dependency index is well enough to an aerial NDVI capture. And the interpretation from these is always has been the crop load varies in these spaces or agricultural lands because of the soil water supply. We have reported on this many times. You can turn these color maps into knowledge. As we studied this even further, the soil sensing measures electrical conductivity. You can train these electrical conductivity values to knowledge if you study it over the course of a year, or even if at the end of the season. This can be turned into knowledge to turn this into a soil texture or a soil water content. In the absence of salting or salt influence in these soils, the soil electrical conductivity gives us the volume by volume content of these soils in terms of water, silt, clay, soil organic carbon, clay, and then estimated soil pore electrical conductivity. The decision to reap these soils is not going to be based on how tightly these soils are structured. There are various options on how these soils can be ripped. So this is a wingless and a single rip that's shown in this photo. And there are two straight shanks to the left of this photo here. Again, the soil profile picture here indicates that the soil needs to be ripped in a multiple direction. Each shank will rip about 8 to 10 inches on each sides. As you can see, as the soil is fracturing here, you do need more than one pass to rip this soil in this case to provide the correct breaking of the pan layers in this case. When should you rip? The soil must be drier than the plastic limit. If you have a high plastic clays like we have in the Oakville station here, you have to wait. If when our ripping is done, when the soil is wetter than the plastic limit, the soil is even worse, and it will restrict water flow or infiltration into the soils even further. When the clay layer is below 18 to 20 inches deep, it might be somewhat more difficult to dry these soils. So you may have option to option to use wing rippers, the goal is not to degrade surface structure while improving the subsurface structure. The wing rippers will use these wing shanks that have about a 20-degree angle. What that does is it vectors the force up. So as the soil is lifted, then it will drop off as it's subsoil to increase numbers of soil. There are also vibrosoilers that we have used on our west side. What vibrosoilers does is it will break up old roots. And these have resulted in better root growth and subsequent vine growth on the west side. In this case, the wings of the rippers wipe up and down twice a second, and then they have about a 4-foot breakout per time. But these require very large tractor to pull, about 100 horsepower. The checklist for soil preparation, again, soil assessment. You need to do a grid or a sensing-based assessment. You need to conclude the soil test, addition of amendments based on our soil test results, deep ripping or incorporation of amendments, and tiling if needed. We do see a lot of tiling in coastal areas now, especially in our NAP tables, and some of our soils very poorly. These are bale loam percentages. These are frequently wet soils. They do have high water tables that result in mottling or glaying in clay and clay loam soils. And we do see soil densities greater than one soil. In this case, most vineyards are tiled parallel to the row for convenience. So, rows can be placed. Usually, we will install a tile drain about every 28 to 30 feet. We do put a 2-inch drop, about 1,000 feet of tiles. The main tile drain is 4 inches in diameter, and the mains are about 6 inches in diameter. But these need to be prepared by a tiling engineer. So here's the tiling plan that we prepared for the Oakville station, and it is very well done here. And of course, we had to bring in machinery to lay the tiles in this case. The water factors is our ongoing saga in California. A lot of things have to be considered, such as the water supply, soil texture and variability, slope, aspect, vine spacing, row orientation, trellis, and our projected canopy side. So we developed quite a few vineyards in Oakville in the last 5 years, and we'll come and install the trellis, and then we'll wait for the irrigation to come in and be installed. And that's what is missing in this photo in the inset. At the design stage, we have to define the water application rate. You have to define the maximum irrigation depth based on soil properties. The rule of thumb is to apply the peak daily ET in inches per day in about a 16 to 20 hour set time. You need to size the components from downstream to the upstream, and flow and friction losses along the pipe system have to be considered. There are a lot of people that design irrigation systems, but as you design these, please keep these things in mind that I will show you. You need to consider the flexibility of operations. These are the range of operating conditions. During its lifetime, the irrigation system will be operated under different conditions. The water needs of young vines are small, then it will increase with time. Blocks at different elevations and distances from water supply will need different amounts of water. There will be blocks with different emitters, which results in application rates, and you will have different emitter size due to soil differences. You will have composite systems, different flow rate and pressure. Some people use drips. Some people start supplementing with micro sprinklers in the valley. There are singles and dual lines. There are alternating or solid irrigation sets. These will affect the quantity, pressure, and factor of irrigation. Groundwater level fluctuates and it will decrease with time as pumps wear out. Some things to consider: application rate has to be less than the soil intake rate. A lot of us are using drip irrigation. Not a whole lot of us are using surface irrigation. Our application rate with drip irrigation is between 0.01 inches to 0.03 inches per hour. So if you consider the idealized curve here, starting with the initial rate to a basic rate, we should be able to do this with drip irrigation very well. But then again, you have to consider your soil type for a maximum application rate at inches per hour, depending on the slope. Most vineyards will have a 0 to 5% of slope as they're prepared. So as for siltier loam soils, our application maximum rate is going to be between 0.2, 0.3 to 0.5 inches per hour. Second rule of thumb is the maximum applied water in inches has to be less than the water holding capacity of these soils. So this is from a textbook. This is the water holding capacity range and averages. For medium texture soils, very fine sandy loams and silt loams, we're looking about 167 millimeters per meter of soil profile. But as the soils get finer and finer, the water holding capacity of soils increase. So a lot of us are aware of this fact. There are quick methods to appraise this method. You can use tension infiltrometers. These usually will have a 20 centimeter base, and they can you can measure these in the field as you're developing these vineyards, or if you're redeveloping a vineyard. This is something I would consider to appraise before the vineyard is redeveloped or developed. There are various methods, of course, to assess maximum water depth and irrigation. Of course, a lot of us are using drip irrigation. The efficiency of application of drip irrigation is between 85 to 90%. DG max refers to the water depth of irrigation. This is calculated by a maximum daily ET multiplied by the irrigation efficiency, irrigation frequency divided by the efficiency of application. So as an example, micro irrigation systems are typically designed for the lowest cost to deliver the peak ET of water needs in a 24-hour set. Better is if it's delivered in 16 to 20-hour sets. So the time of irrigation is going to be equal to the DG max, your maximum depth of irrigation, divided by the application rate. This is also going to be equal to the depth of irrigation by the soil intake rate, which has to be less than the application rate. There's also a soil-based calculation of max of depth of irrigation. In this case, we have to consider the water holding capacity of the soil. You have to consider the management allowable depletion in this cases and a percent wetted area. And then again, the efficiency of application based on the selected irrigation method has to be considered as you're designing these systems. You can convert the water depth that you calculate to water per plant. This is going to be equal to the water depth that you apply in inches per day based on crop spacing. This is from a UCANR publication, evapotranspiration in inches per day by a crop spacing table. You can, pick out the, water depth and crop spacing from this table to derive a water volume in gallons per day in this case. A calculation example from a mature vineyard. This is fairly become a standard. This is somewhat of a, dense planting at a 5 by 6 foot spacing on a VSP trellis. Irrigation system in a single drip line. Rooting depth we assume to be about 5 feet. Effective rooting depth is about 70% of this at 3.5 feet. Wetted area is about 25% of the allotted space for the vine. In sandy loam soils, this is what we calculated. So we're management allowable deficit rate is about 0.8 inches per feet. Our maximum gross irrigation depth to apply in this case is about 0.8 inches. So this turns out to be about 15 gallons per plant for that week. So a lot of these numbers make sense to what we normally apply at the peak of the season if you work through these sheets carefully. Typical flow rates and pressures as you're designing these systems. Drip and micro sprinklers will use about 0.3 to 30 gallons per hour with an operating pressure of 20 to 35 psi. Micro irrigation emitters like the button emitters, micro irrigation emitters require about 7 to 12 psi. Filtering and delivering the water to these emitters on flats require about 15 to 25 psi's. Filters are critical. They require about 15 to 25 psi. If you're back flushing, they will require about 30 to 35 psi to operate properly. There are energy requirements that you need to consider as you're putting in these vineyards. It'll take about 1.3 horsepower per acre foot of water to provide 1 foot of lift. And of course, we have various fuel sources that can drive these pumps. And then I have listed the energy units to lift water per foot. You can see that there's a very wide range of costs with these, but most of us are operating these with electricity. So here's a calculation. If you're using a diesel to pump this water over a large pressure head, the cost for drip irrigation to apply an inch of water is about $2,690 if you're using a diesel, assuming an average price of $2.50 per gallon. Some energy related considerations. Of course, if you're using electricity, there are off-peak hours and peak hours for agricultural consumers. And this starts at a weird time. And a majority of the pumping happens between 7 a.m. to 5 p.m. But the off-peak hours are between midnight to about 11 a.m. So if soil intake rate and our water holding capacity allow, the application rate can be increased to reduce the irrigation set times and benefit from tiered energy rates for these to take advantage of these rates. So here's a checklist. Have a professional system evaluation at least every 2-3 years. Distribution uniformity and application rate will change over time. Know your system application rate. These are key for scheduling irrigations, even for new vineyards. Time to run the system, water to be applied, and application rate has to match. Systems have to be monitored periodically. With new vineyards, you're not going to be applying a whole lot of water. A lot of water will be sitting in these black tubes in the hot California sun. These require maintenance constantly. You need to be checking for leaks, back flushing the filters, chlorinating for organic material for periodic and continuous maintenance. acidifying, cleaning, or replacing clogged emitters for and other components is going to be important as these vineyards grow. We have a publication, Maintaining Micro Irrigation Systems. This is freely available. You can download it from this link. And some of these are available in this publication. Clogging is the main cause of poor system distribution uniformity. Main causes of clogging include suspended material in irrigation water, of course, chemical precipitation as we apply fertilizers, biological growth, root intrusion, and some soil ingestion also happens in vineyard emitters. These are the types of clogging that we see in new vineyards. The action and remedial action is listed. And then I have provide a list here for algae, for hydrogen sulfide, for iron bacteria, iron precipitation, and manganese precipitation, and for slimes. You can look at these lists after the talk is over and see which ones are appropriate for you. The last thing I'm going to talk about is the production factor. Of course, this requires mental readiness and fiscal resilience, cultivar and rootstock choices, plant spacing, row direction, manner of operation, mechanical versus conventional, and of course, a choice of a trellis and conduction system. A lot of movement in the industry now to move towards some sort of mechanization in vineyards. These are the most common systems of trellises that are installed in California. There are three that lend themselves to mechanization. These are the vertically shoot position systems, quadrilateral systems, and single high wire. There's a lot of benefits to all of them, depending on which region you're producing in. There are desired dimensions for end posts and wires using mechanically managed vineyards. For single high wire, VSP, and quads, the post diameters will change. For quads, we use a larger diameter posts that are a little bit taller. For quads and single high wire, we use double spaded end posts. The cordon wires are usually between 10 to 11 gauge for quads, 11 gauge for VSPs. Single high wire does not use any catch wires, but VSPs and quads will use them. The irrigation wires are usually a 13 to 14 gauge wire in this case. There's a shift towards non-positioned conduction systems, and this is by the desire to avoid overexposure of the fruit. The high quads and single high wires do that by making distinct canopies. And in this case, we are able to distribute the temperature on the berries uniformly if we are able to place them at a 45-degree angle from the true north. Here's a checklist for trellis selection. There are desired conditions and corrective actions for plant uniformity, plant condition, trunks, cordons, bearing spur density. Usually for mechanically managed vineyards, it's going to be adequate if we have about 7 to about 11 bearing spurs per foot of row, or 21 to 42 spurs per meter of row. Pruning mass is the telltale sign that these are balanced. We're looking for about between 0.6 pounds to 0.8 pounds per foot of row of growing. I think this is where I will stop, and I can take questions if we have time left. I have not been able to see my chat screen because I'm on a single thing.
We have about 10 minutes for questions, Con.
Okay. Yeah, I'm using a single screen with a slow internet connection. Oh, there are Q&As.
If you have a question, go ahead and type it into the Q&A.
No, we have time then. All right, I can queue up the next presentation and I stop my share. So I have one question. Have I had problems with using EM38 and close trellis spacing? No, we have not had that problem. We have built a sled, and most companies use a similar sled. So this sled suspends the EM38 about 15 centimeters, about 6 inches above the vineyard floor, and it's drawn in between the rows within this sled. So we have not had any problems using it in small spacing. So the smallest spacing we have used is 6 foot in Tokolam Vineyards behind our station here. The question is, does clay soils affect the quality of the vineyards? I know I was going to get this question. So clay soils does affect the quality of the vineyards. What we have seen with the quality of the vineyards with clay soils is that they are more difficult to extract water later on in the season. Although they do have water stored in them, it has been more difficult to extract it. So clay soils will actually stress out the fruit or berries stronger than loamy, silty, and sandy soils. Under irrigated conditions, they have performed better. Next question I have is stakes on the vine or off the vine for mechanical harvest. Yeah, many ways. There's more than one way to skin a cat. If you're running small runs, like a third of an acre rows, roughly about 160 vines, then you can do like a stake at every 3 vines, and then train them up. But for production of, I shouldn't say production, for large scale vineyards, we do post a stake at every vine for a single high wire or for a quad. We do use a beveled stake. These are super stakes, and they are beveled so that they will deflect the wind, because a 2-mile row will become a sail very fast. So we do use a stake. For VSPs, we have used a pencil rod for every vine if we're setting them up for mechanization. For our vineyard, Jerry, at Oakville, I have used bamboo in between line stakes to train them up. And when we mechanically harvest these, we are removing the bamboo, so it's not going to get into the bin or the gondola. So the question from is, under bad water quality problems, such as high salinity contents, what would be the best approach to fix the problem to prevent vineyard quality damage? Sodium damage or chlorine damage is quite common in vineyards. Of course, your best line of defense would be a passive defense, not use that water or not plant there. But if it's unavoidable, like we do, we use various approaches. We do a groundwater recharge. We will push the salts down with canal water. If that's not available, rootstocks are your only option. There are some rootstocks that are commercially available that a colleague of mine in Fresno, George, is working with. 1103P so far is the one that has been somewhat tolerant to saltier conditions, but these are under development by Andy Walker, which he might talk about tomorrow. Yes, I can share the full reference for the Catalyst paper. This also has the videos, etc., for the mechanization or installing these things, doing the practices. Question is, do you recommend staying away from clay soils, and what's the ideal way to deal with them? Well, I mean, it's unavoidable unless you're farming on the beach in Santa Barbara. Soils will have a portion of clay in them, but clay soils can be dealt with tiling them to drain the water away with them, or we have used repeated gypsum applications to make the water infiltrate a little bit better in these conditions. But soils are very diverse. It's sometimes unavoidable to have these clay portions of these soils. Okay. How are we doing on time, Caroline? Should we queue up the next talk?
We have 4 minutes until the break. There is one more question in the chat. What is the most ideal pH for soil?
Well, it depends on what you're growing. If you're growing something like a American cultivar, like a Norton in the Midwest, something between 5.5 to 6.2 pH is ideal. But vinifera grapes, they grow within a wide range of soils between 5.5 to 6.8. But you want to be as closer to neutral as possible, not to have a lot of problems with mineral nutrient uptake, such as iron chlorosis or lime chlorosis, like I showed you earlier. Another question is how much annual gypsum you would need to help with a clear lake clay soil. I don't know. I'll have to ask someone. I don't know every soil.
Con, I think that's it for questions. So perhaps we should go ahead and take a break until 10:30.
Okay. All right. We'll put up the sign. Okay. All right. Okay, break time. I'll mute my thing.
Hey, Caroline, are you back?
Yes, I'm here.
Andreas is here as well.
Yes, I'm going to go ahead and turn off the screen share, and I'm not sure if Karen is there, but well, we can hand it off to Andreas if he's not.
I'm back. Okay. All right. Our next speaker is Andreas Westphal. He is based at the Kearney Agricultural Research and Extension Center. He's a faculty member at University of California, Riverside. And he does work on nematodes in vineyards. So he will be talking about management and control of them. So thank you very much, Andreas, for doing this program with us. So I'll turn it over to you.
Yeah, good morning, everybody. Thank you very much, Khan, for the introduction. I'm sorry that I, for other obligations, had to miss some of the presentations given this morning. I openly confess that. So I understand from the chat and questions that there are some very interesting questions coming along on nematodes, and I'm sorry that I missed that talk, but maybe there's an opportunity at other times to talk to the speaker about that. So I wanted to go back a little bit, and since you heard about it already, what a nematode actually is, what plant parts it infects, what the host status of different plants is, how we can approach a rootstock development, because clearly if the genotypes that we plant into an orchard, into a vineyard, wouldn't even be infected by nematodes, that would be the, well, best situation. But since we haven't obtained that yet, fully at least, we do still need chemical suppression to manage our nematodes. I wanted to also introduce, although it's not that novel anymore, anaerobic soil disinfestation and how we could make it work in our hands for nematode suppression. And then in the end, I would like to give an outlook how we can possibly combine pre and post-plant application of nematicidal compounds. So nematodes are microscopic roundworms. Many times we think about plant parasitic nematodes foremost because, well, those are the ones that become most obvious to us in that they damage plants. And they do that as obligate parasites that they consume nutrients that otherwise we could use to produce yield. Now, they, for the most part, the ones we are concerned about are ones that infect the roots of our host plants. Due to the climate conditions in California, other nematodes are fairly rare around here, but we need to be aware that there are also other ones, namely foliar nematodes, that cause quite some damage in other areas on greenhouses, for example. Now, you may have heard something similar this morning, but just to write home the point, most nematodes are really marine nematodes. So those are that live in the open water, and they are probably totally understudied because they well, are very hard to get by, and they are at very low concentrations in the environment. Then about a quarter of the nematodes out there are free living. So those are very important ones, and I'm sure you heard more about it today. They are very important in nutrient cycling, in that they can feed on bacteria, they can feed on fungi, and that way are very important in the nutrient cycling in the soil. About 10% of the nematode species known are plant parasites. So those are the ones that we mostly focus on today because they are causing damage to our crops. But be aware that about 15% are also animal parasites. So when we say, for example, oh, our puppy has a heartworm, that's actually a nematode causing that damage. Or when we say somebody has worms, we also talk about nematodes. So they can be pretty nasty creatures in that way. Also in here, because I understand that you heard about food webs in the soil, there are nematodes that can feed on other nematodes. They have created some structures in their mouth part that allow them to capture other nematodes and consume them. That would be one issue of biological control, yet those nematodes are very difficult to study as they are difficult to raise in numbers. Now, plant parasitic nematodes, mostly, namely root knot nematode, can be very deforming and challenging to crops as seen here. While celery is maybe not suffering so much from the qualitative parameters here, but only quantitatively, where there's just nothing that grows that could be sold. Other crops like table beets or carrots, of course, are severely damaged and also qualitatively very much in combat with such an infection. Now, while in the annual world, people are still lucky, it's always green on the other side of the fence, they, for the most part, cope with one severe nematode infestation in their fields, if there is such a thing. In the perennial world, we almost always cope with several species at the same time. So we never have the luxury to just focus on one nematode species. That is very critical because our management options, namely also development of resistant rootstocks, has to consider that, because resistances against one plant parasitic nematode does not confer resistance to another. Having that in mind, here you see the dagger nematode, this big one here, and then a ring nematode that many of you know, and root knot and root lesion nematode. And those nematodes can infect the plant at very different life cycles, so or life histories, we should say. To your very left, you see the root knot nematode that invades into the root tissue, makes what we call a feeding site, after which it doesn't move anymore, so it becomes what a nematologist calls sedentary, so it just stays there. Finding resistance for this is, quotation marks, fairly easy, because you just need to disrupt anything of this molecular cascade happening here that allows the nematode to feed there. Once that's interrupted, the nematode, as it is no longer mobile, will die because it doesn't obtain any nourishment. So that's fairly, call it easy for that. Other nematodes of importance in grape is Tylanculus semipenetrans, which is a citrus nematode. This nematode does not embed itself entirely in the root tissue. It stays on the outside and makes this really long neck that allows it to feed within the root tissue. And even less associated with the plant are the ring nematodes. So they are feeding on the plant. Yes, they have a preference and host plants that they like to use, but they stay in the free soil for most of their life history, for all of their life history actually, and only feed from the outside on root. So if you will, there's somewhat of a gradient of how tightly associated a nematode is with the host plant root, from very tight in the root knot nematodes to rather loosely when we talk about ring nematodes. So resistance mechanism, as you can maybe imagine from here, will be quite different and difficult to combine into single rootstocks. That's why folks have worked for so many years in trying to obtain nematode-resistant rootstocks. Now, nematodes are, well, just some scientists like to say like insects without wings and legs, but they do go through a life cycle just like other animals do. So we start off with the nematode egg, shown in the lower left corner, which already contains nematode juvenile. They're triggered by root exudates, comes out of the egg, invades the root, infects it. This is now root knot nematode. Remember, this is very tightly associated nematode with the roots. It goes through this very intricate interaction with the plant where it releases material into the plant that allows the plant to change its cells to make these feeding sites, and then goes through several molds to then produce a male and a female nematode. The female, after fertilization, pretty quickly starts to produce new eggs that it lays into this egg sac. The cycle can, under California conditions, take only close to about a month in summer months, and you can imagine that population densities can build up fairly quickly on the susceptible host. And you can also see in the schematic that the name-giving knots or galls that this nematode causes are increasing in size as the cycling through this life history continues. So this is very obvious to the eye. Other nematodes are more difficult to ID. What is also very important in perennial crops is, in namely grape, that nematodes are very deeply distributed in soil. Of course, wherever there's a root, there's also the opportunity for nematode to feed on. And to our dismay, the nematodes do that. So I brought here a soil extraction from a vineyard soil where you see per depth from 0 to 1 foot, 1 to 2, 2 to 3, and so forth, down to 5 feet, you see the distribution of nematodes in there. And yes, a ring nematode in this particular vineyard, we only found it at shallow depths. That is not indicative that it always must be like that. It just so happened in this vineyard it was. But pin nematode, you see that the highest population densities can be even much deeper than that, at 3 to 4 feet depth. We saw the greatest incidence of them. Whereas citrus nematode was low in population in this vineyard, very low to be of damaging concern, to be honest, but it was present throughout the soil profile. Now, this is very important for our management options, because depending on what tools we use, we need to see that we reduce nematode numbers as much as we can to greater depths, especially in pre-plant options, which we're going to talk about in just a minute here. We do need to reach these populations because pre-plant options typically are very expensive. You just get once in the lifetime of a vineyard to do that, meaning before planting. And so you need very high efficacy for that. So we are challenged with the task to treat nematodes 5 feet deep in soil, which is not an easy endeavor, as you of course know and may imagine in addition to that. So having said that, how can we generally try to cope with nematode problems? As already mentioned, one thing of course would be host plant resistance. That would be very favorable of our vines that we plant in the field don't even get the nematode species, but we also talked a little bit about it, how difficult it is to find a genotype that can withstand different nematode species at the same time. And in chemical control options, we need to distinguish between pre-plant and post-plant option, mainly because though these may be similar chemicals, some considerations are a little bit less important in the pre-plant situation, namely phytotox, because we can use a chemical and wait a long time if necessary before we plant a crop. So there would be no damage from this chemical, which otherwise could possibly happen. In a post-plant situation, of course, we need to maintain our plant intact and undamaged so that we can, of course, foster flourishing growth. Now, if we look into rootstock development, nematologists are well, kind of strict, I guess, in the definitions, but there's a good reason for that. We really distinguish really clearly between resistance and tolerance. It's a little bit complicated to get after this, but the resistance refers to the fact if the nematode can reproduce on a genotype or not. Can it reproduce? Well, then the plant is susceptible. Can it not reproduce? Then it is resistant. This does not say anything to plant growth in response to this infection. That would be characterized by tolerance. So a plant that does not tolerate being intolerant to the damage will respond to nematode populations to very severe damage and very little growth in response to that. Whereas when it is tolerant, it can withstand these infections and grow happily ever after, so to speak. So why is this so critical? Because there can be plants that, yes, they are resistant, but they are only such because they are highly intolerant. So they pretty much give up making any roots by the time they get infected, and that allows the nematode to no longer reproduce, but also the plant doesn't grow anymore. So that's not exactly what we would like. We would like something that is resistant, not allowing the nematode to reproduce, and yet grow happily ever after, which is a very difficult thing to find, especially if we are aiming at several nematode species at the same time, because all these parameters cost energy that the plant needs to get from somewhere. Now, how can we go about doing this? Well, we are lucky that we could join forces with Andy Walker, and so he provided with some elites of his that we planted the offspring into field or microplots. We inoculate with field populations of the different species, best in mixes, to simulate what would happen under real life conditions in a vineyard. We measure the plants and nematodes for at least 3 years, which is an ongoing process, of course. And we try at the same time, since these are oftentimes experimental rootstocks, if we would get a scion to take on these rootstocks. And from there, we could make further selections and maybe propose some for broader testing in broader commercial sized rootstock trials. Now here's an example how something like that could look. So we have here the second year in the microplot trial, so in culverts that are 2 feet in diameter inserted into the ground, filled with test soil. We have 3 different population mixes in here from the vineyard. We have one which we collected from near Livingston, one we collected at Arvin, and one we collected at Selma. All 3 soils had root knot nematode and ring nematodes in there, and here we show population densities of these different nematodes on selected rootstocks. And here's our Chardonnay as a susceptible standard. Freedom is what we thought would be more resistant, but apparently some of the root knot nematodes can reproduce on there. We have Salt Creek on there, which is already better controlling nematode numbers. Leaky 5C, 1017A, some of you may know, and then we have one GRN from Andy and several other experimentals that did not allow reproduction of root knot nematode after 2 years. Now, that of course was very promising, but when we now looked for ring nematode resistance, the picture has changed quite dramatically. Although these are not high numbers for ring nematode, yet there are differences in there, and you can see that our otherwise susceptible standard is now also low in numbers, and that many of our experimental compare unfavorably similar to that susceptible standard. Only GRN1 is doing okay and showing no reproduction, even from field soils with ring nematode population from different areas of the state. Now, that alone then brings up to the next problem. Yes, we want to have something that really can do everything, meaning it can control nematodes not being infected, and then we also wanted to take a graft, which here as an example after first year after grafting, I exemplified here how well the graft takes were, one being 100% of the grafts taking, and well, 0.5 would then be half of them only taking. And you can see that Chardonnay doesn't like to be grafted, at least not to the wine grapes that we grafted on there. GRN, unfortunately, is not doing all that well, and some of the other experiments are not doing all that good neither. As Leaky 5C in the study was doing really well. So just to summarize that, it's very easy to say where we want a genotype that doesn't get infected by as many nematode populations as are out there, nematode species that are out there, but those things also have to be graft compatible in the end. So it's a really long going process to find a rootstock that is really favorable for well, great production. Now let's change gears a little bit to chemical nematode suppression. And as we talked already, there's some hope for pre-plant option where we have some fumigant nematicides. Currently, of course, it's 1,3-D or talone. We have metam sodium, and we have other ones that slowly enter the market. Some of them slow to receive registration, I suppose. Also, that is a very long going process. And then we have non-fumigant nematicides and newer chemistries that's coming online. That is, some of it is already registered, and some of it is awaiting registration. Now, in pre-plant applications, as I already mentioned, we need to treat as much soil volume as we possibly can, because we only get this one option to do that, and this one opportunity well, needs to do the highest efficacy, because nematodes remaining can resume infection of our crop pretty quick. Post-plant applications are more favorable in that sense that they allow for repeat applications, but I mentioned the scare of phytotoxicity already, so then plant needs to be able to withstand these applications. And we, of course, are always concerned about the residue in our harvest products, which limits our application amount that we can put into a certain crop. Now, we went through a whole testing series to see what pre-plant options may be valuable for us to test further. We did this with the help of the nursery industry, who of course has the highest channel to get nematode-free soil. And so we used a microplot setup that I'm going to show a picture of in just a minute. We used water drenches equivalent to 6 acre-inches of water, because the materials that we are testing are not true fumigants, so they need to be delivered to the site of action. We determined the nematode survival. In this particular case, we planted to pre-stratified nemagot seed. Sometimes we have also used plugs, so young saplings of it. And we incubated that under field conditions, determined the nematode penetration after half a year and a year again, and then continued monitoring those plants through plant growth. So we had a whole bunch of treatments in there. I'm not going to go through all of them in detail, but just as an idea. So there are a few materials that are okay at low labor rates and surprisingly effective. Other ones, what I here call high volume materials, are old chemistry in some way, in that they need very high amounts of active ingredient to be highly effective. And then we had our controls, and including also talon in there. So these are the microplots I have been referring to a few times already. So these are culvers 2 feet in diameter. We can put our irrigation system in there, and we can use this volume to treat a known volume of soil in there, because these are inserted 5 feet in the ground. And so we have our test soil in there. We know very well how many nematodes are in there. So it's a very nice contained system to go through a screen like the one that we have been conducting here. Now, when we did that, you see here water control. In this particular thing, we use Pradenius vulnus as our nematode example. Populations are fairly low. It's only 5 nematodes per 250 cc of soil, which of course would already be too high for nursery situation. But the point is that talon 2 reduces nematodes to non-detectable levels, as did some of the experimental things in here as well. But you see, when you go with experimental A, which we applied at different concentrations, that yes, even these concentrations were affording some nematode suppression, whereas the one of experimental B, well, it was mediocre at best. Still some more hope than experimental C that really did not afford any nematode suppression. And here's a good old whited, some of you probably still know was also not faring very well in here. Experimental D was doing okay at different concentrations. And then we get further out here where there are chemicals also involved that probably will never make a registration in California for various reasons, or are forwarded by others now to be further used for pre-plant treatments. So with this, we went to the first screen to see what ones we can use. So we find that some of the fumigant and non-fumigant nematicides do offer some promise, while some of them weed out pretty quickly, at least for these very harsh conditions where we try to eliminate the nematodes altogether. We also see that for a commercial crop, meaning something grown for yield, we probably need a combination of treatments and maybe also need post-plant applications to protect the crop. Now I wanted to briefly change gears to anaerobic soil disinfestation. So I'm talking here about the process that I initiated together with Dan Klipfel, where we use carbon-rich substrate incorporated into the soil, we place irrigation lines on top of the soil, cover the field with TIF, bring the moisture to saturation, and then keep the soil moist for about a month after this initial severe watering. And after that time, we can remove the soil, the film from the field. We have to moisturize it so very heavily because that's necessary for the anaerobic conditions to ensue within the soil matrix. And so for this study that I wanted to present to you, we used 4 different substrates. Some of you have maybe heard or read about anaerobic soil disinfestation. One of the big problems is its price, because materials are placed at 9 tons to the acre before incorporation, which already from the amount creates some logistical challenges, but also rice bran, which in our hands seems to be working the most effectively throughout the different contexts where we tried it in, is quite expensive. Molasses is much cheaper. Tomato pomace is still cheaper, and mustard seed meal is actually much more expensive and maybe not long-term sustainable for an anaerobic soil disinfestation process as we do it here. Now in this study, these materials were applied to a field that was naturally infested after a crop of prunus. We did these studies in August because summer months turned to be out to be the best time of the year to do something like that. We treated them in August, planted our crop in November when it was safe to seed it. So we used pre-esterified Nemagard seed. We monitored the emergence in late winter of the following year, of course. We determined nematode root penetration, and we continued monitoring the plants. And this is what actually happens in the soil environment. So here's the measure that ultimately all we need to know about is the more negative it is, the more anaerobic a soil is. And here just some example. Our control stays anaerobe, although the soil is also watered, yet it stays anaerobe at 18 inches. But in the other soils that received these different amendments and incorporation, plastic lines, plastic cover, soil conditions became anaerobe after different time series of the initial application, but you did get this extended time period of anaerobic conditions in the soil. So you can imagine that such a method is really only fit for pre-plant treatment, because we couldn't do this in an orchard where we would fight anoxia, which would of course be damaging to our crops. So doing this, we can see that the anaerobic conditions develop quite quickly under each of the substrates, so all of them were fit for that. ASD treatments in August reduced Parientis vulnus pretty much as well as the soil fumigation with tallow did, which is a pretty big deal because this was measured down to 5 feet in soil, which shows that we're on the right track there. Top weights were measured, and rice bran had a sustained more vigorous growth of the test plants in there. Now, in grapes with different nematode species present than in almond orchard, there may be adjustments necessary. What I mean by that is that we could not measure good penetration of old roots by these anaerobic conditions. So we are still in the process of examining what the conditions need to be under vineyard conditions, because we know from Mike McHenry that grape roots can stay alive in soil up to 8 years. And as long as a grape root is alive, nematodes can also harbor this root and can continue to survive from there. So that is not a total detriment, but it means that we may have to do some adjustments, and we're working at currently on these adjustments, but it's too early to fully talk about them just yet because data are just coming in. Now, from all these studies that I presented here, it's pretty much one big screening effort. I just show a few of the trials that we did. But I showed you this trial with 15 different treatments in there. And I just went through there and rated plant growth and root lesion nematode survival in the roots. And so a plus was very favorable, a minus, well, negative, so not effective. And you can see among all the treatment that we checked out there that, yes, foremost Salibo at a 2x rate, that was the experimental A, and nowadays we are allowed to talk about which product it is. That was very favorable in supporting plant growth and also reducing root lesion nematode numbers in the roots. And it did that in multiple trials. So this is certainly some keeper. Then other experimental, yes, plant growth was okay, but nematode suppression was not given. So we had to, we discontinued testing them in this context again. Experimental C was even more frustrating in that in multiple tests, so we didn't give up on the material right away. We did give it a second chance, but yet it wasn't really performing to the standard that we needed in here. Same thing provided, that kind of fizzled out. This experimental D is a very interesting material, although it's a high volume material, it needs high amounts of material being added to soil. And we're not so sure how well that is acceptable. Also, we are currently working on new methods to apply this more effectively, because currently we also need to apply it very similar to anaerobic soil desalinization, so drip irrigation lines under plastic top. And then this experimental E and F, those are also some high volume materials. Some of them also highly effective for other reasons. They will not make registration in California, and unfortunately we had to discontinue testing them further. You also see here that our ASD with widespan or also with other materials fared very well. So we do continue working on our anaerobic soil disinfestation, and in different contexts in almond, walnut, and also to some extent in grape, we are trying to figure out if we can simplify the application for this experimental D and also for the anaerobic soil disinfestation, and how we can optimize treatments with Salivro that sometimes can have mixed effects depending on the soil environment where we use it, although in general very favorable. So there are fumigants and non-fumigant materials out there. Salibro was highly efficacious, but the zero tolerance as needed in the nursery, as we could prove in experiments since then, is very hard to obtain with it, if not impossible. We had this one high volume material out there that did repeatedly have very high efficacy, so we are excited about that one. Try to improve application protocols to get the cost down, because currently, quite frankly, it's just too expensive to put all this material out there. It's logistically very challenging, so there's a lot of work necessary on that. Anaerobic soil disinfestation was extremely effective in our hands. It was as effective in eliminating nematode population from soil, similar like soil fumigation with mesobromide or talon. So that's exciting stuff, but there may be limitations of that, as I mentioned, when you're following vineyard with a lot of old grape roots, you may run into trouble there. So having said that, we wanted to apply these findings into a true vineyard establishment trial. So we followed a nematode-infested vineyard. We drench applied our materials that we were interested in 6 acre-inches of water. We then overplanted the plots with multiple grapes so that we can destructively sample after 1 year. We reapplied our treatments as post-plant treatments annually. We monitored the nematode numbers, and we also looked from growth and plant growth and destructive sampling. Now in there, these are the numbers, and here the key nematode was really pin nematode that few people give a lot of credit for being damaging in crops. But if you can look at the numbers here, you saw that especially the one experimental material, which is a high volume material, reduced nematode numbers, and also sustainably did that, because this is from the second year when an additional Movento treatment was put on Talon C on this high volume material. So you did get an additional reduction of nematode numbers out there. And in the first year after these treatments, so I'm jumping back right now, we saw that plants in these plots were growing much better than in other plots, especially this experimental A was not a good formulation of a material, and it caused some growth reduction, as we will see more severely in the following slides. The second year, C, this experimental A, although non-significant, but it's still lagging behind all the other treatments, and only our chemically very intensive treatments improve plant growth some. Now in the third year, the experimental F really outperformed the other plots, even Talon C, when it was combined with Movento. But as I mentioned earlier, unfortunately, there's some limitations of its opportunity to be registered in California. And well, that's unfortunate, but that's in science or in research. You just sometimes set on a horse that then will not make it to the finish line. So having said that, so there are pre-plant treatments for this experimental materials out there. But what we also learned from these studies, and being somewhat new in perennial crops, less than 10 years, I have to confess, it really drove for me the point home that it is so critical that we start our perennial crop planting properly with as clean of a vineyard soil as we possibly can. That is part of our planting procedures and how we plant our vineyard. We could also see phytotoxicity issues, and the one experimental treatment I pointed out, the vines never grew out of this trouble. They continue to look very poorly. So that's why this kind of studies that we do at Kearney are so critical, so that we can find out about these things before we ever go to a growers field with treatments that ultimately just hurt the crops instead of helping. Now, if there's time, and I believe there is, I wanted to briefly talk about a new nematode in California. Not that we wouldn't have enough problems already as it is, but there is the peach root knot nematode we found a couple of years ago on root knot nematode resistant rootstocks. Now, when we say that in California, we mean the southern root knot nematode population. So Hansen 536 and Nemagard should be resistant to it, but a tree should have at least looked like this in the orchard, and instead it was looking poorly like this. When we took roots out, we found a lot of galling on the root systems out there. So that was quite a shock at first. We found out that this nematode can also infect grape, so it's not without significance for you, but we so far have foremost found it in different almond orchards. CDFA and the Ag Commissioner, or CDFA, rates this as an A-rated quarantine pest, meaning that we should be vigilant when we find it, that we put such a vineyard or orchard under quarantine so that we don't spread the nematode further. And the first line of action should actually be eradication, as long as we find it only in very limited acreage. And that's currently the case. So that's our recommendation, that at least no soil or roots are moved from such a vineyard or orchard to other sites. Now, peach root knot nematode, since we found it here, it was also found in Florida. It was first described, and that's a typo that's of course not found twice in Florida, but many more times. This was supposed to be California. It's found in California, in Georgia, and also South Carolina. So it is of significance. The significance is that root knot nematodes in general have a very wide host range. We're not sure if this one is maybe foremost infecting perennials, but also in annual crops. Colleagues of mine at UC Riverside in the quarantine facility down there could find out that it also infects a lot of vegetable crops. The significance for the perennial world is that it does overcome root knot nematode resistance against the southern root knot nematodes. What it does in grapes overall, there's a long ways to go and learn about that, I guess. There are some rootstocks proposed out of Florida that are also available in California, but those exemptions that those would be resistant to peach root knot nematode don't really hold up. And so there are breeding populations of prunus out there which have promise, but we are just beginning to work on that. And since it is a quarantine pest, we can only really work in our quarantine facility that we have on the UC Riverside main campus. We can only work on it there, and we have been doing so on a very small scale just to get some sense in how our California rootstocks would respond to that. And yes, in some of our breeding materials, there seems to be some stuff that is more resistant to it. When I say our, I mean, of course, the breeder cooperators of mine and the ones that I test for nematodes here. It was found in Merced and Kern County, so very far apart from one another. And it is likely to have been present for a while, because at least at the Merced site, we found it on very old orchard trees too. So it's probably just the find. It's not that it's now suddenly going to spread wildly. It's just that the finds may increase over time as people become aware of this nematode being out there. What should we do when we find it? Well, we should work with the Ag Commissioner, although that sometimes is uncomfortable for us because we don't like to do that maybe. But it is important to get them and us involved so that we can quickly find it, so that we can find out how widespread that is. Do I, as a nematologist, have to focus resources on it, or is it such an exotic find, let's call that I don't really worry about it, but better focus on the nematodes that we know for sure are already here. So that's why this is very critical. There was a pest alert published on that, and we go through different trade journals to publish more information on this, but this would perhaps be a good starting point for you if you wanted to learn a little bit more of the background about it. And so with this, I think I used up most of the time that was allotted to me today. But before I conclude my talk and maybe look at the question and answers section on the slide, I would like to thank those folks on here. This stuff is done in collaboration with many different people from different departments at Riverside Davis, with CDFA collaborators, with of course the farm advisors and several individuals, my research team, the Kearney crew who works on it, nurses who graciously support me with a very crazy requests at times, pest control companies who make some of the studies possible, and then funding agencies down here that I don't really want to name singly, but I certainly acknowledge and appreciate the strong support we get from that. So with that, I want to thank you for your attention. And if there's still the time for me, I would like to once check if there's anything in the question and answer that I should answer now, or should I do that later? I'm not quite sure. So.
Well, thank you, Andreas. That was very good. We do have time for a couple of questions, I believe.
Mm-hmm.
And we have quite a few questions, actually. So the first one is from Chile, actually. What adjustments need to be done with ASD in order to be more effective in grapevine nematodes such as Zephaniema and root knot nematode?
Well, the adjustments are that we know Zephaniema, we haven't tested against it because it wasn't necessarily present in the vineyard that we put out and we did this. The bigger issue is the survival of old grape roots, which could harbor things like root knot nematode. And we are trying right now, if we were to kill the vine before we even consider ASD, if that would help in giving ASD a higher chance. But in a way, we are still in the middle of those investigations, so it would be a little bit early to make a recommendation based on that.
Okay. Next question is, what do we know about the impact of all these fumigants on soil microbiome health and diversity?
Well, of course, the more broad spectrum biocides will perturb the microbial populations in the soil. Typically, microorganisms come back pretty quickly after this, and it is then a different microbiome than before the application was made. Many times it's a favorable one for plant growth. Yeah, and so of course we disturb food webs, but all those things do reestablish fairly quickly.
Yeah. The other question is, have you done any evaluations with the material Diteria DF? It has Myrothecium verrucaria as the active ingredient biological.
Well, it's an aged material, and so I know it has been around for a while. I have in previous lifetimes, I have tested that. Actually, I was still a graduate student at the time, and I wasn't convinced that it necessarily would need to be included in my grape testing.
Okay. Fair enough. Next question, is sweet potatoes the link for the RK floridensis?
Can you repeat that? I didn't hear the one word you said.
Is sweet potatoes the link for the RK root knot floridensis?
Well,
I think that's the question.
Yeah, well, I think it goes to this anecdote, which unfortunately was developed when we first found it in Merced, that somehow the interplanting of sweet potatoes in a almond orchard had brought in floridensis from somewhere, but there's no scientific base for that. Also, what in my mind speaks against it is that in the same orchard where we found this nematode first on very young tree. There were old trees right next to it, and we found it on the same as much. And they, to the best of my knowledge, had no intercropping, no nothing of that. So I detest when there's too quick things thrown together. Sweet potato is, of course, a very good root knot nematode host. I don't question that. But I think to make that connection would be not proper because there's no data basis to justify that.
All right, the question and answer panel is still open, but I'd like to thank Andreas again for joining us, for taking the time to update us with this very informative talk. So I'll turn it over to Cindy Crone, who's the new IPM advisor in the three Tri-County region. Oh, I'm sorry, 4-county region now. Sonoma, Napa, Mendo, and the Lake County. So she will talk about ecobiology of the grapevine red blotch vector in commercial vineyards. So I'll mute myself. And
thank you, Con. So today I'm going to talk about the ecobiology of grapevine red blotch vector in commercial vineyards. And specifically, I will be talking about the three-cornered alfalfa hopper and their relationship with vineyards. So early on, in 2016, three-cornered alfalfa hopper was identified as a vector of grapevine red blotch virus in a greenhouse study. And from that, we moved forward trying to figure out what is their relationship with vineyards. There was very little that we knew starting out. And so we knew that the scientific name is Spicillus festinus, three-cornered alfalfa hopper. It was shown to be able to vector grapevine red blotch virus in a greenhouse study. Is native to the United States, so it is not an invasive species. It was studied predominantly on alfalfa, peanuts, and soybeans in the southern United States, where it was a pest of those crops. And it was previously considered only an incidental pest of grapevines. And this was because you could see girdling late in the season of petioles and shoots in the vineyard, but it rarely was ever associated with economic damage. Or extensive economic damage. Very little was known about the association of this insect with grapevines because of that, because it hadn't been considered an economic pest previously. So starting out, we didn't know a whole lot, right? We didn't know why they were in the vineyard. Were they always in the vineyard? There was a lot of things that we needed to learn about the biology and behavior of this insect and their association with grapevines. And the first thing I set out to look at was what plant species serve as hosts of three-cornered alfalfa hopper in vineyards. And so what I did was test weed species that were commonly found in vineyards that I found in Oakville and surrounding vineyards. And this was the results of that first study. And as you can see here, you have your scientific name, the common name of the weed that was tested, the family in which the weed was in, were girdles present or not. So when girdles are present, you know that three-cornered alfalfa hopper is fed, but they do not need to girdle in order to feed. So if girdles are not present, we used the data of percent survival. Previous research that was conducted with Newsom et al. In 1983 showed that the adult three-cornered alfalfa hopper can live on water alone for 12 days. So percent survival in this graph is at 14 days, with the belief that if the adult was still alive, if any of the adults were still alive at 14 days, they had to have fed on the plant material. So in the situation where you have girdling is no, such as buckhorn plantain below, but you have a percent survival of adults at 14 days, you know at some point the adults had to have fed. And then the other thing we were looking at is were nymphs, did nymphs emerge? So were eggs oviposited in which nymphs emerged from the plant material? We did not follow through to see if the nymphs actually were able to become adults on the plant material. When I'm talking about a reproductive host, I'm talking about, did nymphs emerge? Were they able to lay eggs, and did the nymphs actually emerge? And so what we found is that Spanish clover, dandelion, bird's feet trefoil, common groundsel, and field bindweed were all feeding and reproductive hosts of the three-cornered alfalfa hopper. And we found that Kentucky bluegrass, buckthorn plantain, and wild carrots were poor feeding hosts. And you can see that by the low % survival after 2 weeks. They were not reproductive hosts. Sorry, my screen just moved. They were not reproductive hosts, and they're not likely to be significant in maintaining the populations of three-cornered alfalfa hopper in a vineyard when there's more suitable hosts present. And when you're looking out in a vineyard, you usually have an array of different weeds and even sometimes planted cover crops that three-cornered alfalfa hopper can feed on. And what we saw also too is that in this, the sharp point fluval and Bermuda grass were not feeding or reproductive hosts at all. So the next group that we looked at were cover crops that are commonly planted. And what we found is that all of the cover crops in the family Fabaceae, and Fabaceae is legumes. So all the legumes were feeding and reproductive hosts. Which was supported by previous research on the peanut, soybeans, alfalfa. Again, those are legumes, that legumes are the preferred host. So this supported that information even further. And what we found also too in those cover crops, only found that mustard and California red oats, out of the ones we tested, were not feeding a reproductive host. So with that, we wanted to know, in these original tests, the question was, can they feed and reproduce, yes or no? So they were caged onto a plant, not allowed to move off that plant. They didn't have a choice. So the next step is doing the choice studies. So we took the weeds and the cover crops that we knew were feeding and reproductive hosts from these no choice studies, and then put them in a tent, as you can see here, where the adults were allowed to freely move about the cage, feed as they wanted to, reproduce, lay their eggs wherever they want to, and they had a choice at that point. So trying to look at in a situation similar to being out in the field, it's not the same, but similar as far as you can in a lab setting, greenhouse setting, make it, give them choices and see what were they going to prefer. So, and what we found from this is that when we were looking at the vineyard weeds, purple vetch was a standard in every run that we did. We know that they reproduce on purple vetch. We raised them on purple vetch. So we had purple vetch in every run as a standard to compare it to. So the vineyard weeds included 3 species in the legume family, and 1 in the daisy family, and 1 in the morning glory family. And what we found is that the 3 legume plant species had significantly more nymphs emerge than did the plants in the other 2 families. And you can see this indicated by the letter A and A are considered statistically the same, where B and B are statistically the same, but different than purple vetch, Spanish clover, and bird's trefoil. The next group that we looked at were 5 cover crops, and they were all in the legume family. But we did see a reproductive difference that existed. So the 2 vetch species, so your purple vetch and your woolly pod vetch, were preferred over the 2 clover species, the white Dutch clover and the subterranean clover. So even within legumes, it seems, this was the initial looking at, that vetch seemed to be preferred over clover. And then we moved on to another set of 5 cover crops, 4 in the legume family, 1 in the grass family. Purple vetch and bell beans had significantly greater nymph emergence than did blando brome in the grass family. And in each of these 3 groups tested, the plant species that had the greatest nymph emergence were all in the legume family. And again, this further supports the hypothesis that the plants in legume family are the preferred host of three-cornered alfalfa hopper. So we have 2 non-reproductive cover crop hosts and that we found, but growers need more options. 2 is not enough to say, oh, limit it to these 2. So I moved on and I tested 15 more commonly planted cover crops, trying to find other options and see if there were other options that for growers to use that were not feeding a reproductive host if growers chose to do so. So here's the list. Again, it's the same setup: scientific name, common name, family, and then girdles, nymphs, and percent survival. And so this was the list that I chose of commonly planted cover crops in California. And what we found was that buckwheat, Trius triticale, annual ryegrass, purple vetch, berseem clover, and Zoro fescue were all feeding on reproductive hosts. And in that list, Piper Sudan grass, winter white ryegrain, barley UC 937, and daikine radish, perennial ryegrass, they were feeding hosts, but not reproductive hosts. And so these species can support feeding, but they don't allow three-corner alfalfa hopper to increase in population. So they don't serve the same role as a feeding and reproductive host in an orchard vineyard setting. And then what we found is that orchard grass, creeping red fescue, fawn tall fescue, hard fescue, and California poppy. And I know California poppy is not planted as a cover crop, but it was one that came up commonly as a question from growers because California poppy grows just wild everywhere, it seems, in California, obviously state flower. I tested that also. And so we found that these following cover crops were not feeding or reproductive hosts. So those are the ones in yellow added to possible, if there is a concern of a red blotch of spreading in your vineyard and you want to reduce that possibility, possibly by choosing a cover crop that they cannot feed or reproduce on. These would be good options. So we went ahead and we did the preference test again, and we chose the cover crops that had shown to be reproductive in the study. And again, vetch was preferred over clover. So as you can see here, we have the purple vetch with an A and the berseem clover with a B. But the 2 legumes, so berseem clover and vetches are both legumes, are preferred over all the remaining families. So as you can see here with the C, that the vetch and clover are preferred for all the others. And there was very few nymphs found on the buckwheat here and the Zoro fescue, as you can see, very few. So although it was possible, it did not seem as an ideal option for three-corner alfalfa hopper. And then no nymphs were found on triticale and annual ryegrass. So although triticale and annual ryegrass, they're capable of reproducing on those cover crops, when given other options in the field, they did not choose to feed or reproduce on them. So moving forward, another question that was commonly asked, are grapevines a reproductive host? And so what we did is that we took adult three-corner alfalfa hopper and we caged them on different sections of a grapevine, dormant cane, on green shoots, and then the apical shoots. And what we found that, well, first of all, you can see the photos. Yes, they did lay eggs in when the adults were caged onto the apical shoots and the green shoots. They did lay eggs, and here's photos of the eggs embedded in the bracts and the buds, and also this is all little slits in which eggs are oviposited into the petiole. So yes, they did lay eggs into the grapevine material. But what we found was that they did not feed, the adults did not survive for 2 weeks, and they did not reproduce on dormant wood. They only laid eggs and reproduced and stayed alive as adults on green shoots and on apical shoots. And that is where we have our data. So what we found, so here across is apical shoot data. On the bottom, we have the green shoot data. And in this graph, in blue is the number of first instars. So Three-cornered alfalfa hopper has 5 instars, and basically that's 5 immature stages. It goes from egg, first instar, second, third, fourth, fifth, adult. And so as you can see in the graph, the first instar is in blue, and this is the number over time, and the second instar is in orange. Now, in this period of time in which we did the study, you would expect adults to have emerged. You would expect to go from egg, first, second, third, fourth, fifth instar to an adult in this time period. But what we found was that we did not get any instar past the second instar. So there was a high nymph mortality and no nymphs past the second instar, which they didn't progress in development as you would expect them to. And our results suggest that grapevines are not an ideal host for feeding for three-cornered alfalfa hopper nymphs. And so, as you can see here in this one, what this graph over here is showing is that here's in blue is the total number of first instars. And in gray, that's the opaque portion, was the portion that were dead. So when I opened up the cages, I counted the live and dead because they both emerged. But I documented what percentage were actually dead in that cage. And as you can see, a large portion in both the green shoot and apical shoots of both the first and second instar were dead. They were not alive, which also supported the understanding that yes, they can lay eggs in green shoots and apical shoots, and the nymphs emerge, but they don't seem to be able to complete their life cycle on grapevines, and it doesn't seem to be a good enough host to keep them alive for a long period of time. And so this doesn't, although it sounds odd, it's actually not too odd, because research prior in apples had shown that nymphs emerged from the buds of apples and fell to the ground and finished their life cycle on the planted alfalfa, which is a legume, in the orchards. And this was done back in the early 1900s, but it is a documentation of that behavior of emerging from where the eggs were oviposited and dropping down below and finishing a life cycle on ground cover. So it should be said that three-cornered alfalfa hopper are reported of overwintering as adults and eggs in the southern United States, but it's undetermined whether three-cornered alfalfa hopper actually lay eggs in grapevines. Now we know that they're capable. We don't know if they actually are doing it. That would need further study. So then going on with these other questions, how many generations does three-cornered alfalfa hopper have? Do they overwinter in the vineyard? What is their spatial distribution? All these questions that are listed here were more questions that we needed a larger study to try to address. And so what I did, this is Oakville Research Station. This was the block that I sampled. I sampled every other row, because every other row was tilled under. So I sampled every other row that had ground cover. So it was 26 rows, and the perimeter around the irrigation pond. It should be noted that I sampled the ground cover and not the canopy. So throughout the whole season, I was sampling the ground cover with a sweep net. And also there was an irrigation ditch running along this side here in black. And that's going to be important a little bit later when we're looking at spatial dynamics. But yes, there was an irrigation ditch along the east side of the block. So this is 2 years of data. We started collecting on March 16th of 2016 and ended on March 18th of 2018. When we first sampled, our first sample date, the adult three-cornered alfalfa hoppers were already in the vineyard. So we missed the immigration this season, and that is why we continued. We were able to capture the immigration in 2017, and why we continued into 2018 to capture that immigration. So this is 2 years worth of data, and the first tree hoppers were found in the vineyard on February 15th of 2017. So this is February 15th, and this is February 1st of 2018. Adult tree hoppers were found in the field prior to bud break. This red arrow right here was bud break. So here we even had them in the field before bud break, but here we were able to capture them immigrating 7 weeks prior to bud break. And at this point in which we were sampling, we sampled 6 weeks and bud break still had not occurred. So what this indicates is that three-corner alfalfa hopper is attracted into the vineyard by something else. It is not coming into the vineyard for the grapevines as a primary host. It's coming in well before because of the previous research where we showed that they did not survive on dormant wood. We know they are not feeding on the dormant wood. So if they arrive in the vineyard 7 weeks before bud break, they're feeding on something else. And it's not the green material of the grapevine that's attracting them into the vineyard. So some other interesting things that we found is that flowering coincided with the first infield generation jump. So what happens is that the overwintering adults arrive in the vineyard and they lay their eggs in the ground cover and they proceed to die off. See this this period of time that we're seeing right here in both years. It's a depression in the adult population. This is pretty normal. Adults spend all their energy in the winter time trying to make it through the winter, and they just have enough to mate, lay eggs, and then they die off. And then the new generation emerges, and that is the generation that will go through the season, over winter, and come back the following year. So this depression is not abnormal, and it actually is a point of time in which the nymphs are developing. And the issue with so the green little dots that we see here are actually when we found nymphs. The issue is that when nymphs first emerge, they're they're about 1 millimeter, and they're clear, and you can't find them in a sweep net. They're too small, they're too translucent, and not as noticeable. We actually didn't pick up nymphs until the 4th or 5th instar. At that point, they have a different coloration, you know, either bright green or they have a mottling brown, and they're chunkier, larger, that's very noticeable within the leaf litter of a sweep net. So during this period of time, they're developing, but sweep nets don't really pick them up. The first day that we picked up a nymph was the first day that we saw the jump in the adult population. And again here, the first time we picked up a nymph was the first time we see a jump in the adult population. So what's going on is the adults are overwintering, they're coming in the field, they're laying their eggs, they die off. This is when the nymphs are developing. This is when the first infield generation emerges. And again, we see that same pattern here. The first infield generation emerges. In 2016, we saw 2 different generations. And in 2017, we only saw 1. I will also say that during this period of time, we had major fires. Although you would, if there was another generation, you would expect it here, and it didn't appear. With that, so the next step that we did at the same time in that same field that we were testing, we would count girdling in a subset of vines in Oakville Research Station. So I took the data of the sweepnet data that you see here in green. And in blue, we have our data of girdling that we saw, because there was a question by growers, well, do they girdle all year long? I only see the girdles in the fall. Do they girdle, is it early, are they transmitting earlier, but I just don't see the girdles later? Like, what's going on? And we didn't have an answer. So we mapped girdling in a subset of the vines. And as you can see here, that girdling was first documented in June, starting in June. Was our first documentation of girdling. And the girdling continued over the season. This period right here where we saw this huge depression was during those major fires in which the sun was blocked out. Actually, I couldn't even gain access to Oakville Research Station because of evacuation. And then the girdling went back up as soon as the air cleared and the sun was able to get through. The insects went back to behaving normal. So what we noticed is that, again, the first girdling started in June that we were able to document. And and research happening at the same time. So Sinowitz et al., so it is Mark Fuchs' group in Cornell. They were looking at Spicillus facinus and whether they were testing positive or negative over a season. And the first positive three-cornered alfalfa hopper was documented in June. So although what I was looking at was different than what they were looking at, it makes sense. If the adults are starting to move up into the canopy to feed in June, that is the first time that they would be acquiring the virus by feeding on an infected vine. And this is the first time that another group is also finding three-cornered alfalfa hopper starting to test positive, which was in June. So when asked, when is the girdling starting? It's starting in June, and that is when it seems, from the data we currently have, when transmission is starting also. So what we have here is 2 years worth of data. So we have 2016 and 2017. These are the Sadie red-blue plots. And what this all means, what you're looking at is red. So as the hue is deeper, so the red indicates aggregation of the insects. And so where the insects are aggregated in a higher population. White, any kind of white areas in between is randomness, where there's really no pattern that they can find. And then blue is uniformity. And what that means is uniform as in they're not finding any three-cornered alfalfa hopper. So it's uniformly, as it gets darker blue, uniformly not finding three-cornered alfalfa hopper. And so what we found is that aggregation occurred on the vineyard edges. So statistically, when we tested the vineyard edges compared to the center, the vineyard edges statistically had more aggregation than the center of the vineyard. And primarily also on the eastern edge. So see on this side, this is the eastern edge, and this was the eastern edge. Remember I had mentioned that irrigation ditch that ran on the eastern side of the edge? So there seems to be, from our experience, some type of association between water and populations of three-cornered alfalfa hopper. Now, we don't know why. We don't know if it's actually the water, or maybe there's more reproductive hosts growing near water, and therefore they are more likely to be found, or more legumes growing wild near water. We're really not sure why, but this is something that we do see this pattern. It's the reason why I sampled around the irrigation pond, because over this time of learning about three-cornered alfalfa hopper, we did see this pattern of an association where if you wanted to go to a new vineyard to sample, you would look for the legumes, you would look for the water source, and you would sample there. Another thing that was very interesting about this 2 years worth of data is you see how in 2016 in the north, there was definitely a lot more aggregation in the northern part than in 2017. In 2017, remember, blue is uniformly not finding three-cornered alfalfa hopper. So interesting enough, in this north of this block, so would have been up here, this year they had, the field was fallow. So the field was fallow, they planted it with bell beans. They at some point tilled it under. But early in the season there was bell beans well into springtime. The second year they had replanted this vineyard in the north with new vines, and mustard was throughout the rows of this northern portion. Remember, mustard was not a feeding or reproductive host, where bell beans was. Now, is this proven by what I'm showing you? No. This is something that it's anecdotal. We know, we see it, we're like, oh, there's a pattern. What does it mean? What was going on around it? And it's a way of possibly explaining what we see in this aggregation and lack of aggregation from one year to the next in this area. So that was an interesting note. So what if you're a no-till vineyard? You plant legumes as a cover crop for nitrogen, and you're concerned about grapevine red blotch disease in your vineyard. So looking at, okay, what are some other options? And that's where I went with the research. When I continued on working with the USDA in Parlier, I was going beyond like, okay, what else can I be looking at? So you can kill the cover crop with steam if you're no-till, but it's not commonly done, and I don't know really the expense of being able to do that, if it's cost effective or not. But I then thought of, well, can we use biocontrol? You know, what insects, and there had been research previously done testing big-eyed bug, damsel bug, and minute pirate bug on the different life stages of three-cornered alfalfa hopper. But what about all the biocontrol that you can buy from Insectary? What about what we have easy access to? Have we tested any of those? And it hadn't been tested. So I moved forward and I looked at 6 commonly easily accessible purchase biocontrol from different insectaries. So we did the convergent ladybird beetle, adult, the millibug destroyer larva, which is here, millibug destroyer adults, assassin bug nymphs, brown lacewing adults, and green lacewing larvae. And so what we found, so how did we test it? There was two arenas. The first arena was a petri dish in which we had a little cut of green bean so that the nymph could feed on something, but that was it. It was the immature stage or adult stage. We tested all 5 instars and adults separately. Against, so you had 1 prey, 1 predator. And so the bell bean cage is the petri dish cage is the most artificial. The question at that point is it within what the predator sees as prey? Are they capable of killing, consuming this insect or not? Are they interested at all? And then we did bell bean cages. So is a potted bell bean, individual bell bean, that's covered, one prey, 1 predator, and they were allowed 24 hours to move about however they want within there. And now that situation mimics more of a real world situation. Not completely, of course, but it is the best you can do in a greenhouse situation. It allowed the prey places for them to be able to hide, and it required the predator to be able to find the prey. So with that, what we found that there was statistically no significant predation of the 5 immature stages and adults of the three-cornered alfalfa hopper by the millibug destroyer larva, millibug destroyer adult, and the brown lacewing adult. But what we did find when we were looking at assassin bug nymphs, so what you're seeing here above is the petri dish and the bell beans. So you'd expect higher significance in more things being significant in a petri dish because it's easier for the predator to find the prey than a bell bean cage. And basically, if mortality was positive, so above this 0 line, and the 95% confidence intervals did not pass this 0 line, then we knew that the mortality in the presence of the predator was considered significant. And I marked the one significant with an asterisk. So as you can see, this is the 1st, 2nd, 3rd, 4th, 5th immature stage, or also known as the instar, and the adult stage. What we found is that there was no significant predation of any adults in any of the things that we tested. So any of the insects that we tested, nothing really went after the adults. So we focus on the immature stages and assassin bugs. So in first instar of an assassin bug, there's significant predation on 1st and 2nd instar in a petri dish, but no significance in the bell bean cage. Now, assassin bugs are great generalist predators. We know this. And the reason why I tested 1st instars is because we could only get eggs from the insectary. So I tested what a grower would be using. And it's possible that if we tested later instars, 2nd, 3rd, 4th adults, we might have gotten different results. But we tested what was commercially available. And we were not able to find any kind of significant predation in the bell bean cages. So we moved on and we tried convergent ladybird beetles. These are very common. You can buy these at your local lumber store, right? And what we found is that first, second, third, fourth instars, significant predation in petri dishes. But we only found significant predation in first and second instars in the bell bean cages. Now, this makes sense, and the reason being is that convergent ladybird beetles, their preferred hosts are aphids. Aphids are about the size of your first and second instars, so it makes sense that that would be a preferred prey size for them to go after. There are some issues with possibly using convergent ladybird beetles as biocontrol, being that they have an innate they innately want to fly. So the minute you release them, they want to fly west. And that's because they're harvested in the foothills, and their natural tendency is when it starts to warm up to fly west towards the crops. So when you release them out in the field, that is still it's innately in them to fly. So they're known to not really remain where they're released, which wouldn't make them really great biocontrol for your current vineyard if you were to release them. So that's something to keep in mind. And so then the last one that we tested was the most promising, green lacewing larvae. And there was significant predation of all 5 instars in petri dishes and in all 5 instars in bell bean cages. Now, with that said, future research is needed to see if the biocontrol can play a role in reducing three-cornered alfalfa hopper populations in the vineyard setting. There are other things in play when you release biocontrol into a vineyard setting. You have alternative prey that they can choose that they might prefer over three-cornered alfalfa hopper nymphs. They also have predators that will be coming after them, which we don't obviously isn't mimicked in the bell bean cage that we tested. So it's not necessarily transferable, and further research would need to be done to be able to say that there is a possibility that green lacewing larvae can reduce populations in the vineyard setting. But I will say that this result was definitely promising, and this seems like a pretty good candidate to move forward and test in the field. So what did we learn with all this information? Legumes are preferred hosts of three-cornered alfalfa hopper, and that's weeds and cover crops. So control of leguminous weed hosts is important in thinking about reducing viral spread within a vineyard. Three-cornered alfalfa hopper can reproduce on grapevines, but adults do not survive or reproduce on dormant wood. So we know that virus transmission is not expected during the grapevine dormancy. And nymphs do not complete their life cycle on grapevines, so controlling the ground vegetation that serves as a host could help reduce the population. Three-cornered alfalfa hopper arrive in the vineyard in late winter. So we saw one year they arrived February 15th, the other year February 1st. That's pretty early in the season that they first start arriving into a vineyard. So sampling for three-cornered alfalfa hopper is needed earlier than would be expected. And when I say sampling, it's sampling by sweep net. Sticky cards, although will capture the random three-cornered alfalfa hopper, they do not represent the population that's in the vineyard. And I've done this when I was doing sweep netting on a height of a day, I think I caught over 70, and I had none in my sticky cards for that time. So sticky cards are not the ideal way to sample for this insect if you want to go ahead and sample. The overwintering population arrive in the vineyard before bud break. And since they don't feed on dormant wood, we can deduce that three-cornered alfalfa hopper are initially attracted into the vineyard by other feeding and reproductive hosts. So controlling the vegetation that serves as hosts in your vineyard could be the next step and the best way of moving forward with that. Overwintering adults arrive in the vineyard to feed, lay eggs, proceed to die off. And that's where I showed you that indentation where the low population was happening. And this is a time in which nymphs are developing and the most susceptible stage for insect for control. because immatures don't have wings, they cannot disperse widely. They are on the plant or near, they can only, the first instars pretty much don't even move from the plant in which they emerge from. The second, second and third instars tend to stay at the base of the plant. Fourth and fifth instars will move, but not long distances because they have to walk, crawl. They cannot fly, they have no wings. So if you till under the cover crop, if you are a till vineyard and you till under the cover crop before the nymphs are able to complete their life cycle, then you're removing the feeding host for nymphs and they will die. If they don't have a host to feed on, they'll die from starvation. So this large reduction in the first infill generation would be expected to have an overall seasonal population reduction in that immediate area. Of course, you do have adults that can come in, you have adults that can leave, but it is a matter of trying not to attract them into your vineyard in the first place, if at all possible. And then, if you're going to till under your cover crop, timing it at the ideal time in which the immatures are developing. The adult population is really low, immatures are developing. If you remove their feeding hosts, they die, they starve. There's no way for them to continue their survival. So as the ground cover dies off in the vineyard, three-corner alfalfa hopper adults move up into the grapevine canopy to feed. And we documented when weeds started to die on the ground, and that's when we started seeing the movement up into the canopy by girdling. So this is the likely timing in which the virus acquisition and transmission occurs. And that's when you looked at Sinowitz et al.'s data showing that they just started to test positive in June. So, and that's the same time in which we started to see girdling up in the canopy. So it appears to be 1 to 2 generations per year. So if you're lucky in that. If you have a large reduction in the first infill generation, then you may have an impact on the degree of virus spread overall. Fewer insects, the idea is fewer insects would equal reduced vector-mediated spread. Spatial distribution is aggregated, and what that means is more samples are needed. So if you have aggregated distribution, you need to take more samples to be able to find the insect. It's easier to miss them in your sampling, and therefore you need more samples than expected. Edge effects exist and possibly attraction to water sources. So what does this mean? This can really help drive. If I go out to a vineyard, I know exactly where I want to go and sample. And this is where I want to sample: near water sources, near the vineyard's edge, adjacent to riparian areas, areas with leguminous cover crops or weeds, and or if high grapevine red blotch disease occurrence. This is where I go to a brand new field and they say, do I have this insect? That's where I go to look to find them. So that's a great way of, focusing. Like if you have a short, don't have a whole lot of time to sample, where do I go? You go to these areas and try to find them there. So although further research is needed supporting green lacewing populations in the vineyard, may contribute to the reduction of three-corner alfalfa hopper populations in the vineyard. So the idea is that if you have fewer nymphs, you'd have fewer adults that emerge. But again, further research really is needed to show that what we saw in the greenhouse is actually applicable to the field. And with that, I will finish my presentation. If anyone is interested in reading more about the research that I've conducted, these are the papers in Journal of Insect Science, Journal of Economic Entomology, Journal of Enology and Viticulture, and PLOS ONE. And I don't know, do I have time, Con, for questions?
You do have time, and we have the grower panel at the same time. So
Okay.
I think we can take the Q&As for you, and if there are any general questions for our other panelists, keep them coming in the Q&A. So the first question that popped up on my screen is from one of my former students, Mr. Pansare. Yes, the slides will be made available. So anyways, let's see. There are some questions that were not answered previously. So here's one. Are there cover crops to avoid that act as hosts of nematode pests? If anyone's able to answer that.
Are you asking, I'm sorry, is this for Andres?
I don't know who it's directed to. Um, but next question would be if there are any legumes which do not support the alfalfa hopper.
Every legume that I've tested has been a feeding and reproductive host. I have not found any documentation in previous research of a legume that was not a feeding and reproductive host.
Yeah.
And that doesn't mean that it doesn't exist, because we have not tested every single possible legume out there. But I did test quite a few legumes. And previous research were on peanuts, soybeans, alfalfa, peas. These are all feeding reproductive hosts and problems.
Next question. Cover crops don't host the virus, correct? Only grapevines at this point. I assume the virus being grapevine red blotch.
So there has been different groups that have looked and tested cover crops and weeds and other plants. And from my knowledge, currently they have not had any positive grapevine red blotch virus positive results from weeds or legumes. What they have received positive is Vitis vinifera and wild grapevines growing in riparian areas have tested positive. So with that said, wild grapevines in riparian areas could possibly serve as a reservoir.
That's true. Oh, as a non-host tika, I'm gonna cover crop in the vineyard.
I'm sorry, Con, you broke up a bit.
It'd be a non-legume. Question is, which cover crop would you use as a non-host tika permanent cover crop in a vineyard?
Well, if you go back, grasses seem to be the most promising, but grasses are not always, ideal. It really depends. When a grower asks me, the answer is, well, why are you planting your cover crop? What are you trying to get out of your cover crop? Because although, as you can see here, orchard grass, creeping red fescue, fawn tall fescue, hard fescue, and previously it was mustard and California red oats. These are all the ones that we tested where we did not find feeding or reproduction on. But it really depends on why you're planting the cover crop in the first place. So grasses are not ideal for all situations, especially in areas where, water's scarce. So these are what I've tested so far, but that doesn't mean that there's not more out there that are good options too. They just haven't been tested.
Okay, another tika question. Why does the transmission only start in June while the tika moves into the vineyard before bud break?
Yeah, because so three-corner alfalfa hopper moves in the vineyard before bud break, but I had shown that they don't feed on dormant wood. So they only would start feeding, or they're capable of feeding after bud break. But there's plenty of hosts on the ground and usually preferred. So if you have a legume compared to a grapevine, they prefer the legume. So as long as there are feeding hosts and reproductive hosts on the ground, they're not moving up into the canopy. What we notice is that as the ground cover starts to die off, because that area is not being watered in the summertime and it's hot, it starts to die off, then we see that movement up into the canopy. So that's, I mean, that's what we've seen in research. It cannot be definitively said, like, definitely they don't do it for this reason or that reason. But it does make sense that if they do prefer, if legumes are the preferred host and that's available somewhere in the vineyard, that's where they're likely to be. Until that host is no longer available, then they'll move on to whatever host that they can feed on, a secondary host, or, you know, not as preferred as a legume, just to feed to stay alive.
All right. These next two questions are similar. Do chicken or wild birds eat the tika, or have there been any studies on biocontrol by encouraging birds, such as bluebird with bluebird boxes, to eat the tika?
There has been research documentation. I wouldn't say research. It's been documented in very old papers in which observational studies happened. They've been found in the guts of frogs, toads, frogs that were, you know, feeding on three-cornered alfalfa hoppers, and in birds. So yes, in birds. Now, there hasn't been research to show having a population of this number of birds reduces the three-cornered alfalfa hopper population by this much. These are references in literature made many years ago, before, where observational studies were published, where now it seems that only actual research is published these days. Did you know what I'm saying?
Yeah.
And so these are definitely, but they did dissect birds, and they did dissect frogs, and they did find them in the guts. Spiders would be something good too. You know, spiders are generalist predators, and spiders would feed on them.
Cool. All right, we have a question from Galt. Evan Goldman is asking, will mowing the row meadows reduce the populations of tika as well?
That's a very good question. And actually, Kent Dane, his lab is working on this currently. And their research is not yet published, but they have been looking at this. And I'm excited for when their results come out, because I think that's a very good question. And from my understanding, three-cornered alfalfa hopper can feed at the very base of the plants, at the soil, even sometimes slightly below the soil as it matures. So if you were to mow, there's still enough if mowing does not kill the plant itself, if mowing just reduced the amount of plant material above ground, but did not eliminate the plant material above ground, the immature stages and adults can feed on that little stubble. And it's enough for them to finish their life cycle. But I will say, beyond that, like I said, Kent Dane's lab is actually testing this. And I'm excited for when they are able to publish and release that information.
Okay, so this is another question for Cindy. So the question is, and I have the same question as well, has there been any studies that compare incidence severity of red blotch across different vineyard flood management practices, such as 100% till versus no-till versus legume, cover crops versus perennial grasses, etc.?
Not to my knowledge.
Yeah.
Kent Dane's lab, I don't know if they're looking at disease incidence or not. I do know that they're looking at insect populations of bare ground, mowed and I think just regular,
Yeah.
untended. But I don't know if they're associating that with disease incidence or not. But to my knowledge, I am not aware of a project like that.
No, and we have an active red blotch project with Kent and Anita and Sudi. And this is within the realm of that project, but we're not looking at different species of cover crops. It's tilled versus non-tilled in this case. So, yeah, but no, work has not been done on this topic yet. Okay, next question again for Cindy. When overwintering adults arrive in the vineyard in February, how far could they be flying in from? Just an adjacent property or from miles away?
They are good flyers. They can fly some distance. We don't know exactly how far they can fly, but I do know when doing research in Davis, when they would mow the alfalfa fields a mile away, they would end up in my vineyard. So, and I always knew when they mowed because all of a sudden they'd end up in my vineyard.
Yeah.
And you go down to that, and the alfalfa fields is where we collected them anyway. So there was that association. So I know that Houston Wilson did some work looking at transects into riparian areas and into the vineyard at different distances to see is there movement. And we've spent a lot of time sampling for this insect out in the vineyard and looking riparian areas. And we don't exactly know where they go. There's been previous research done in the South where they found them on pine trees in the winter. But I've never seen them on pine trees myself. And that was not done in California. So we're not really sure where they go. We know that when I sampled, and I sampled every week, even in the winter, I didn't find them.
Okay. All right. Here's a question for Andreas. Are there cover crops to avoid that act as hosts of nematode pests? Maybe
Can you
not.
hear me now?
Yes.
Oh, okay. Well, thank you for that question. Well, similarly, like trouble in our main crop and cover crops, of course, we also need to be aware that there are several nematodes out there. And so you always look to the one shot fits it all, but that's unfortunately difficult. So we have gone through screens of different brassicas. We have gone through legumes. Among the legumes, many of them do support ring nematode populations. So we had a little bit better luck with one of the clovers in there. Rose clover was less supportive under greenhouse conditions, for example. Then we had pretty good luck with rye. Rye has many years been used as a cover crop in root knot nematode infested area for other reasons, not necessarily in grapes. But it is a poor host for root knot nematodes, so it can support minimal numbers of root knot nematodes, so not the greatest host plant to use neither. It foremost really depends on the nematode population that you have in your vineyard, and that may vary from vineyard to vineyard. We are about to complete those studies and write that information up, and we hopefully come up with a decision grid and what kind of nematodes you have present and what crop would withstand that. But there's not one that, or that's the bottom line, I guess.
Yeah, very rarely there's a silver bullet. So let me see. For Christina. Oh, here's another one for Andreas. Aside from good cultural practices, what else can be done to control nematodes in organic vineyards prior to new vineyard establishments and already established vineyards?
Yeah, that's a big challenge to to find solutions there. So I know that there's some organic quotation mark nematicides out there. Not so convinced that they have much of an efficacy. What can help is some nematode antagonistic cover cropping, but we are probably too far off to really make recommendations on that line yet. But there are crops that would allow, at least if it's in vineyard preparation, to grow these crops and try to reduce nematode numbers that way, that they become less of an issue when the organic vineyard is planted. I know that's not very satisfying, but that's unfortunately it's an uphill battle in an organic vineyard to come up with good solutions. But of course, issues like a conventional grower would use too, having a rootstock choice will be helpful. And again, it always comes back to what kind of spectrum of nematodes do I have present and which are the most likely to cause trouble. And then from there on out, go after rootstocks that hopefully are less susceptible than others to the ones that I have present in my soil.
Okay, here's another nematode question. Have you ever looked at crab shell products as a potential control agent for nematodes?
Yes, it's that has been a very hot topic some decades ago, I have to say, where people used crab shells and found results with that. We are looking into that. It may be that if you put crab shells out as, well, pretty much unprocessed form, let's call it that, meaning like as chunks in the soil, it's probably difficult to get a good initial efficacy of that. Numbers are extremely variable, so I'm not convinced that a simple application of crab shells will solve the issue. But certainly there could be potential there. The idea is that organisms in soil that consume the chitin of the crab shell, that those are enriched, and the nematode egg shell has also chitin in it. And the idea is then that these organisms then first, chew up the crab shells, and then out of lack of other substrates also chew up the nematode eggs. The concept is very nice. To really make it work under practical conditions, that's like always a challenge with something that is more biologically oriented.
Thank you. So here's a question for Cindy. We seem to find a more slow wave-like spread of a red blotch from one end of a block to the other, which seems odd for Tika, which seems to be able to travel fairly far distances. Any thoughts? Anonymous attendee asks. You're muted, Cindy.
We should first say where we currently stand, there's only been one insect, three-cornered alfalfa hopper, that has been shown to be able to acquire and transmit this virus. But it was a greenhouse study. It was not done in the field. And there is current research underway looking, and there has been current research and previous research looking at other possible insect vectors. So with that said, there is a possibility that three-cornered alfalfa hopper is not the only vector. And then we are looking at more, a different problem. But I will say that if three-cornered alfalfa hopper arrives in a field and finds legumes and finds a host, they're not likely to move long distances. If they're happy where they're at, they will stay, feed, reproduce where they're at. And so do you see a great difference in the amount of weeds that are legumes or hosts in the vineyard? I do know that when I sampled Oakville Research Station, legumes, there were legumes there. There was burr clover, Spanish clover, and sometimes the random, white Dutch clover popping up here and there, but it wasn't uniform. Some rows had all grass, and I never found any three-cornered alfalfa hop in them. And some rows had like big, bunch of Spanish clover, and that's where I found a bunch. So it's not your ground cover is not necessarily uniform throughout your vineyard, and that could have some impact in how you're seeing it move from year to year. Because if you till under, your your weed seeds are getting moved around, it's possible that that changes from year to year too. Yeah, I don't, there's no, there's so many variables that could be contributing to that, that there's no exact answers. Those are just some of the options of what could be going on.
Thank you. Lauren asks, are there any promising organic post-planting treatments for nematodes? To Andreas.
Thank you for that question. It seems a bit similar to what I mentioned already. So I think it's right now it's very difficult to find something there. So
Yeah.
and the stuff on the market, I'm not so convinced that there's too much benefit in them.
Okay, thank you. So this is a question for Cindy. What would be a good cover crop or a cover crop mixture to repel a three-cornered alfalfa hopper? And would creating a border with it around the vineyard help?
So any of the ones that I've tested that I had listed, that were in yellow, that we're not feeding a reproductive host, those would be the ones recommended. If you're concerned about red blotch and its spread, and giving three-cornered alfalfa hopper a place to feed and reproduce. I'm sorry, what was the second part of the question, Con?
Would you be able to create a border around the vineyard with this?
You can create a border around the vineyard, but it will not, it's not likely to be effective.
Right.
Because you create a border, but you have inside your vineyard legumes of some sort, weeds or cover crops, they're going to just fly past that border into the center where they can find what they're looking for.
Yeah, which is food source.
There has, unless you're asking, can you create a border with legumes around the vineyard to keep them from going into the vineyard, that as a trap crop that you could treat or get rid of. And yes, that's possible, but it is challenging. And the reason being is you are giving them a place to feed and reproduce. And if it's not managed very carefully, you could actually just be increasing your population. And typically around a vineyard is harder to plant because that is where the tractors are driving. The ground is very hardened, it's not normally tilled. So it would be hard to plant and hard to remove.
Well, it'd be hard to irrigate these too later on in the season when they're feeding. Yeah, these border cover crops.
Yeah, you'd have to be very careful with that one.
Yeah, I wouldn't do it. So here's another question for you, Cindy. Do you have an opinion about Sauvignon Blanc being a non-symptomatic host for red blotch and thereby providing a strong source of infection for vineyards that are in close proximity to Cabernet? How much distance would you need between Sauvignon Blanc and Cabernet Sauvignon to provide some buffering?
So that hasn't been looked at yet. So there is no research to give you an answer of this is what we tested and this is what we found. White varieties in general are asymptomatic, and they don't turn red. You get some chlorosis. Some are a little bit noticeable than others later in the season. But what distance this insect, we don't know exactly how far this insect can fly, but I do know that it can fly, over a mile from just experience, personal experience. It's not documented. It's not we haven't tagged the insect and found it a mile away. So because it hasn't been looked at, there's no way of really telling you what distance would be, quote, safe. The further away you would assume would be safer, but that doesn't necessarily mean that it is. Yeah. But any grapevine that's a source is a source. If an insect lands on it, three-cornered alfalfa hopper lands on it, feeds on it, acquires the virus, and then moves away, then it feeds on an uninfected vine, it has the ability to transmit.
Okay, here's a question for Andreas. Will promoting soil microbial diversity suppress parasitic nematodes enough to be an effective control strategy? Any ideas about this?
That's many times we think that from a gut feeling, well, we have more diversity out there and things are going to be more healthy, but that alone will probably not do that. I mean, organisms countering nematodes are highly specialized organisms, and if they are not present just because there's a lot of diversity out in the soil doesn't mean that it also suppresses nematodes. So diversity alone is probably not going to do it.
Okay, thank you. Okay, here's the $1 million question for you, Cindy. Thank you for an incredibly interesting presentation. Has transmission of a red blotch been proven outside of a greenhouse setting by the three-cornered alfalfa hopper?
Not to date, no.
Okay. Another question regarding the three-cornered alfalfa hopper. Can it transmit Pierce's disease?
I'm not sure if anyone has looked at that, but I don't know. I don't know if anyone's looked at that specifically. They're not believed to, but that doesn't mean we really know.
Yeah, I don't think we know.
No, I don't think anyone's looked at that.
I don't want to think about that.
Right.
It just gave me a cold chills down my spine. Anyways, Phil Fries from Yountville asks, does red blotch survive in nymphs from stage to stage?
So no one has looked at that. But if you think about the behavior of the insect, the nymphs are developing on the ground cover. They're not feeding on the grapevine itself. And then how would they acquire the virus? If cover crops, if ground cover, so far to our knowledge, and in situations where it's been tested have come back negative, we don't believe they're reservoir host. How would the nymph acquire the virus in the first place? And the virus is circulative. So in theory, that would be possible, but it has not been tested. And so we really don't know. But is it really likely to happen when you think about the behavior of the insect and where they're located in time?
Yeah. Here's another question for go ahead.
So to respond to, I just got a text to respond. This is a very good point. When you're talking about Pierce's disease and three-cornered alfalfa hopper, Rhonda just, she made a very good point. It's great. Three-cornered alfalfa hopper is a phloem feeder. And so Pierce's disease requires a xylem feeder. So it's very highly unlikely.
Rhonda, huh?
That feeds in two different places. That's a very good point. I didn't even think about that.
Yeah. This is the same Rhonda we know?
Yeah, Rhonda Smith.
Yes. Oh yeah, I remember her from a while back. So another question for Andreas. Is there any research showing the efficacy of Dithera as a post-planting nematicide? Because it was asked earlier as a pre-planting nematicide.
Well, we haven't tested it due to my experiences in earlier lives with it, that we had a hard time finding efficacy with it.
Okay.
So,
All right. Andrew Quaddy is asking to Cindy, other hosts for a three-cornered alfalfa hopper besides the legumes, such as blackberries that might be growing in thickets. And
So it depends on, Andrew, are you asking about a host for the virus or a host for the insect to feed on? As a host for the virus was found in blackberry, but with this is the caveat, but when further tested, was it believed to be a feeding site where the insect originally fed. The virus did not spread systemically through the blackberry, so it would not be considered a reservoir host. Now, if you're asking about a place for three-cornered alfalfa hopper to feed, I have not ever found three-cornered alfalfa hopper nymphs on blackberry. And in the literature, I have not seen blackberry reported. So according to what we know, that would be no. That doesn't mean it's not possible. It just might not have been documented.
thank you. So Andreas, last question is for you. Are there predator nematode species identified that could be a potential biocontrol option to control plant parasitic nematodes?
that's a really nice question. So yes, there are predatory nematodes. For example, the genus Mononcos has some of them, and so they literally seek out nematodes and feed on them. So for that reason, they would be pretty attractive for that. Trouble is that so far people have challenges to grow them in culture because to make higher population densities of them, well, they very quickly feed on one another. So they turn cannibalistic in a pretty fast fashion. So that kind of takes away from that. So predators are maybe a little bit more difficult. What people have tried with varying successes, didn't work all that great in our hands, but people have tried to overwhelm the nematode niche, if you want, with insect pathogenic nematodes, entomopathogenic nematodes. And those can be grown in huge amounts in fermenters, and they can be, they can go anhydrobiotic, so they can be dried down on surfaces. So shelf life is fairly nicely solved. And then the idea is that you pour those into the soil and that they then compete with the plant parasitic nematodes about space, if you will. Personally, it hasn't worked so great in my hands, but that's something that people have been trying. And the predatory thing is certainly an interesting idea because it would be a similar niche that they are occupying. They would have a lot easier time to get after them. So.
Okay, well, I'd like to thank all the panelists today for joining us. If you're registered for day 2, we will see you at 8:25 a.m. tomorrow. So I'd like to thank all the speakers again, Andreas from UC Riverside, Cindy from UCC Sonoma, previously of UC Davis, and Christina, who left to go teach. And also I'd like to thank Caroline and Karen for putting everything together and keeping me on the dot as they usually do. And also my partner in crime, extension, Anita Oberholster. So there will be a quiz to get your DPR credits. So you will receive it from Caroline. So make sure you answer the questions. It'll have all the information for you to get the correct hours. So tomorrow we'll start at the same time again. We will have a weed control. We will have our rootstocks. Christina will talk about the soil health survey that she did in Napa. And Dr. Torres will talk about mycorrhizae solutions, which we seem to be applying a lot. And she'll tell you if it makes financial sense or not. So with that, thank you all for attending today, and I will see you tomorrow.